Robin is there.
Acha. Yeah,
Thank you so much, sir.
everybody
Namaste,
is
sir.
not on this screen. So today is the third day.
Yes, sir.
Robin is visible to me now. Mandalji.
They have to tell about Dr. Dastur. They have to tell.
Yeah, that's what I asked you. Shall we start? And would you begin or I begin? No problem. Or maybe I begin and then you follow the next and
Yeah.
Within 3 minutes, we'll
so
start.
congratulations
Okay.
to the speakers.
The speakers,
So-
Dr. Kajal and Dr. Pramod. Pramod has joined.
Pramod has not joined.
Hello, Pramod. You have joined.
Hello.
So Pramod has not joined. So we'll wait 3 minutes to start the session.
So I shall introduce Kajol. As I
Sir,
said,
as a chair, you have to introduce the speaker, and we will introduce from our on behalf of the society to the chairs. Okay.
Manohar sir, good morning. Namaste.
Good morning. Namaskar. Namaskar.
Morning, Secretary sir.
Good morning, Dr. Marcus.
Yes.
Good morning, Sesa.
Good morning, Sisa. Good morning, ma'am.
Rashmi ji, good morning.
Good morning,
Good morning, sir. Welcome. Nice to see you, sir.
For Deepika's
Yes, yes.
the
She could
nomination
finish.
occurred yesterday.
Okay.
The
Thank you, sir.
notification came to me also.
Okay. So thanks to you, sir.
No,
You helped so much and
You are always
cooperated
welcome.
also. Yeah. It's a really pleasure.
I
sir.
simply felicitated so that, she gets
No, it's
educated
wonderful.
and
Yeah. Some issues were there with her, and then, of course, she could do it. Waiting
what
for
do
the
you
people.
want? Who is going to introduce the speaker?
Sir, speaker to आप ही करेंगे.
By the chairs.
If I have to do, I am glad to do it for Kajol first. No
Yeah.
problem. He has been given the award lecture this time, and I am glad that he's in time, and he is known to me for almost, if I'm right, 20 years. Is it correct?
More than.
More than that.
Be gone.
More than her.
Kajol has disappeared.
Makesh.
Good morning, Devappa.
Good morning.
Morning.
I think we can ask the organizer to give the brief biodata of Dr.
He
Kajal
should be
to
visible on this.
No, first,
Yes.
Finally, he has come.
Kalyan will
So,
begin
So,
the session.
So, ma'am, just we are waiting 1 or 2
Waiting.
minutes because
Yes, sure. We can wait for 5
Yes.
minutes. Nobody. Nobody.
most of you or everybody knows Kajol, and he's a well-known virologist for us, and he has, enough expertise in the field. So
Sir,
I don't need to go in much detail except that he was born in 1967 in West Bengal, and he had a master's in agriculture in plant pathology from BCKV, one of the good institutions, and PhD from IRI, the Mecca, and
sir, we have not yet started the program.
Yes, sir. Solve कर दे, हैं?
এই সেশনের পরে না একটা এরপর তোমার এখানে হচ্ছে এপি স্টেবল গ্রান্টেড প্রেজেন্টেশন না তার আগে সাবনি টিপাটির
I think it's fine.
So a very good morning to all of you. On behalf of Indian Fightball Society and on behalf of organizing committee, I extend a very good welcome to all the delegates of today's session. And I think most of the participants has already joined. And this session will be chaired by two eminent personality in plant pathology, who is very well known to us, Professor S.M. Paul Khurana. So before start the process, and also jointly chaired by Professor Manohar Acharya. So before starting the session, I would just, for the newcomers and for the students, I would just brief about, introduce Professor Paul Khurana. Professor Paul Khurana had postdoctoral research in advanced virology from Japan, University of Minnesota, and also University of Minnesota, USA. And he served at different capacities at pivotal positions of different institutes, including as a Vice Chancellor, Rani Durgavati University, Jabalpur, as well as Director, CPRI, Simla. And he was also founder director of Amity Institute of Biotechnology at Gurgaon and remained as a consultant to CIP FAO, and also has a very vast experience in plant virology as well as biotechnology. And he is credited with 255 research papers, 150 reviews, 25 books, and guided 16 PhD students. He remained fellow and honored fellow in different prestigious societies, including National Academy of Biological Science, fellow National Academy of Agricultural Science, and he also traveled very widely to different international conferences, including in UK, Europe, USA, Malaysia, and he is really a great inspiration to the society, and he served this society, and we are very privileged that he served the society as a president, as a also in to guided us in a different, you can say, guided us in a good manner for the society's growth. And we are really thankful, sir, for kindly give your consent to chair this session. And thank you. Nice. Now I am going to introduce Professor Manohar Acharya, again, a eminent personality, stalwart in mycology. Particularly, I would like to mention for the young scientists and researchers and students that Professor Manohar Acharya served Osmania University for 45 long years in different capacities, including as a dean. He served as Vice Chancellor of Oriental University, Indore, and he guided 50 students of PhD and published 640 research papers and 30 books, including that of Springer Publications. And he also served as a chairman, expert member of different, you can say, research supporting systems, government systems like UGC, DST, DBT, and ICAR. And he also National Academy of Science, member of National Academy of Science, fellow, and fellow of National Academy of Sciences. So really, we are thankful, sir, for your kind consent to chair this session, and we are really grateful to welcome you, both of you, chair. So with this, I just request the chairs to start the proceedings of today, which will be an award lecture, and I think the chair will introduce the speakers of the awardee. Thank you very much. Paul Khurana, sir.
Yes, thank you very much. In fact, I thought, that we should save on time rather than introducing ourselves. We will introduce the speaker, which is more important. Even though they are so well known, nonetheless, you have done it, so I'm thankful, and I'm also thankful to Professor Manohar Acharya and to Professor Rashmi and Professor Pratibha, who are very much here. And I don't see eminent Bhisham Pitama, our Professor Anupam Verma and Professor Barnwal. So, nevertheless, Kajol is a virologist. We were collaborators. We even had a common student from IRI to MIT, and she had PhD, Shruti Gudara. And the beauty is, I wanted her to work on citrus, and so was Kajol. But somehow the problem precipitated to cotton, and the work done by her was good enough. Nevertheless, that's the quality or the importance of Professor Kajol, and I'm happy that he supported her a lot. And Kajol, I don't need to go into detail that he was born in 67 at Nadia, and he did his MSc at BCKV and PhD from IRI, the Mecca, and had a postdoc in Gainesville, the one place I could not visit despite my wish. And um he has enough research experience now in molecular plant pathology. He had a very good book, which I have on my shelf here also in plant virology. And not only that, he also came up with citrus fruits and cultivation diseases and their management. So he is good at many things. He can do work and he can write equally well, and then he's very friendly and very kind. Nevertheless, with all this, I congratulate him before the lecture he gives, and the award lectures are a matter of prestige for our fellows, and there are many other things. I'm sure that you would be happy. I can also see Chakravarti Saab now here, and MS Saharan is also there, and very good that many others would have joined. Basavaraj is smiling for something. Anyway, with all this, I would stop here. Otherwise, I can keep on speaking. So may I request Kajol to kindly come up and take 20 minutes, because we have another speaker and we have total 1 hour.
Thank you, sir. Now I am going to share my slide. Now it is visible.
Yes, sir. It is visible.
Visible, and my sound is okay.
Yeah, you are audible.
Yes.
Very clear.
Thank you, Dr. Khurana sir. And I am very much grateful for the Indian Phytopathological Society, our big society, considered me as a person for giving this prestigious J.F. Das Memorial Lecture Award. And I am very much thankful to all the committee members who are organizing this conference in very methodical and very nice manner, and particularly Dr. Robin Gogoi. He, in this COVID situation, he has managed our IPS that is beyond our imagination. And now again, I want to give my thanks and my pranam to our former head, Dr. Pratibha Madam, and our present head and dean, our Rashmi Madam. Always, the both of them are giving me love, just like their son. And just recently, Dr. Pratibha Madam told me, Kajol, I'm happy to see you. Ma'am, why? No, always remember, good thought, good words, good action, then destiny. So giving to pranam to our two head madam, former and head, and Dr. Manocharya sir, Dr. Chakravarti da, Apurva da, my friend Markesh, all many friends, Krishna Kumar also, because when I was a PhD student, Krishna Kumar was the SRF in our laboratory, And then I want to bow my head, Professor Anupam Verma, Professor Chitresh Sen, Professor V.G. Maluthi, Padma Madam, Professor Alawat, Dr. Barnwal, Professor Jain, and many, Dr. P.K. Chakraborty, because when I was in Gainville, P.K. Chakraborty was with me for 1 year. We stayed in the same room for 6 months. So many friends, many well-wishers, many students behind me for this coming, behind me to come in this position and to give my prestigious lecture, J.F. Dastu. Understanding of the sleeping giant virus in citrus and cotton, perspective to economic losses in India, and challenge to battle. First, I will tell regarding citrus virus, because since my beginning, I started my career with biological control, Professor Chitresh Sen. And then I did my PhD on plant virology. When I got the posting in Krida, Hyderabad, again, I had to work on the biological control, integrated disease management. Again, when I went to Kalimpong, again started my virological work. And during PhD, I have done my work, Mungbean yellow mosaic virus. I have lot of work in Mungbean yellow mosaic virus. But in this lecture, I want to focus on 2 aspects, citrus virus and cotton leaf curl virus. Both are the giant virus, sleeping giant in crops of cotton and citrus. Now, citrus virus, it is known that citrus is the most important crops in India. It ranks 6th in production in the world, and it's ranked 3rd after mango and banana in India. And in area, 9.23 lakh hectare, where citrus is cultivated with production of 8.7 billion tons. But if we see in the world, the citrus has $9 billion global industry. The citrus is sadness because it has killed millions of citrus trees in South America during 1940, 50, 60s. And it has caused death of 100 millions of citrus trees. Symptoms is vein clearing, yellowing, vein corking, stem pitting, and ultimately declining. It is transmitted through Toxopteris citricida, and this virus is located phloem, and phloem of the plants become victimized due to the infection of the CTV. Now, CTV is monopartite particle, positive sense RNA virus, having 9.5 kg nucleotide length, and it contains 12 ORFs encoding the virus activity, infections, and other. And now CTV has 7 different genogroups worldwide, and it has 7% nucleotide difference among them. Now, Brown, 1920, first time he showed the evidence of citrus virus in the Punjab. Then Nagpal, Kapoor, Vasudeva, they also reported Tisteja in different orchards in Mumbai. Then Narayani reported Tisteja in 1965 in North India. Then Kapoor and then Chakraborty identified various Tisteja strains based on biological specificity and differential host. And Dr. Balaram and Ramakrishnan identified mild strains and severe strains. Dr. Alawar is a pioneer worker in the citrus virus. In India, he detected CTV first time by ELISA and electron microscopy. And he also shown, and Chakraborty also shown, that CTV is similar like the CTV present in US. After that, Manjunath and Roy first time they started the molecular biology of the CTV based on the code protein gene. They cloned, sequenced, and analyzed but it was that work was being done from US. After that, what I have done what we have done in our laboratory, then we study incidence, then molecular diagnosis, characterization, complete genome we have cloned and sequenced, recombination analysis, genetic stability, origin, and evolution. And then we have made some strategy for productions of disease planting material and management. Now this is the survey, extensive survey I have made throughout the India, Assam, Darjeeling, Central India, Meghalaya, Manipur, Sikkim, and you see the decline is the major symptoms caused by citrus virus. Now, CTV infects all the important citrus and its relatives. It has killed 1.5 million citrus trees in India. Accordingly, Dr. Y.S. Alava, Dr. Baranwal, Dr. Poon, Dr. Krishna Reddy, because it is a very notorious disease. Nowadays, this disease is to some extent less because it is the sleeping giant. Anytime that virus can come up and to cause havoc disease like that. CTV incidence has been estimated in India 16 to 75%, but in Northeast India, the incidence is very high, and most of the citrus orchards are wiped out day by day. Now, genetic characterization and variability. We have taken 105 isolates for WRF1 gene, 101 isolates for CP gene, and 20 isolates for the suppressor gene. And with these genes has been amplified with the specific primers, clone, sequence, and analyzed. And out of all the 105 WRFs, isolates of WRF genes, when we have analyzed, we have seen 9 groups worldwide, and India is having 8 groups, and they are having 78 to 99% sequence similarity. When we have studied the core protein gene, then we have seen India is having 7 and world is 8. That means most of the variants of citrus virus presents in India. And we have studied the distribution. The most of the variants are present in the Northeast India and maximum in the Assam and Darjeeling. North India, Central India, and South India, there the variation is to some extent is less. Now coming to number of variants, and after that again we have added another 40 isolates, and based on these analyses, presently in 2020 we have seen that at least 10 CTV variants prevalent in India. Then one complete genome first time has been cloned, sequenced, and biologically characterized. The one of the strain, that's the KPG3, declined strains from the Kalimpong. We have maintained the here, and we have done the biological reaction. Based on the biological reaction, we have considered this strain is the declining strains, and that strain, that complete genome has been amplified, cloned, and sequenced and analyzed. After analysis, we have seen this KPG3 is related to the Israel isolates VT, which is severe. But in our India, this KPG3 is the slow declining. That's why these strains cannot kill the virus immediately. It takes time. Now, recently, total 32 isolates containing 38 complete genome has been analyzed worldwide, and we have seen there are the 7 strains: T36, VT, T3, RV, T68, T30, our KPG3 under the VT group. Now, again, we have studied another 4 isolates from different places and containing the 3' gene, 8.3 kb. And based on this analysis, we understood that Indian CTV isolates are the very much distinct, and all are different. Now, recombination study shows that Indian isolates, when we have compared with the Asian isolate, we have seen Indian isolates is non-recombinant in the 3' ends compared to Asian isolate. That means Indian isolates are very much distinct compared to other. Now, genetic stability, origin, and evolution, we have studied based on the 122 isolates of 100 isolates where core protein gene has been used. From where the 29 originated from citrus aurantifolia, 38 isolates originated from citrus reticulata, and 55 originated from citrus sinensis. In our laboratory, we have used 83 and other area 39. Based on this, the Bayesian skylon plot shows that evolution of the population size of CTV is very low. It is 6.5 into 10 to the power 4 per year to 1.4. That means that the mutation rate is very slow. And the time scale Bayesian shows that the India shows that two major branches were formed to 107 years back. That means 107 years back, whereas CTB was the constant because mutation rate is very low. And the Skyline plot showed that CTB population size remained constant until 2001, an increment of 1 log, and suggesting that the expansion has been started after 2000. What is this? What is going on? It is coming here. And the CTB population display low codon usage bias and higher genome stability. When we have studied the code-protein gene sequence and citrus chromosome and citrus sequence, we have seen the codon usage biasness of the CTB because the similar types of codon used by virus and citrus. When the mutation rate is very slow, that means natural selection is more profound than that of the mutation pressure shaping the CTB population. Now, correspondence analysis and contribution of high-frequency codon analysis using the 102 code-protein genes, it has been shown that citrus reticulata-originated CTB display higher value, indicating that codon uses adaptation to CTB to citrus reticulata host, and this analysis also showed that the citrus reticulata is an evolutionary primitive, and CTB might be originated millions years back from the citrus reticulata. That means mandarin. Now coming to management strategy. Birdwood grafting, raising nuclear seedling, generation of plant through tissue culture techniques, root tip grafting. This effort was made for cross protection and also from transgenic resistance. Now identified the mother stalks, raising the, maintaining the healthy compatible root stock, then grafting healthy sign on root, disease-free grafted plant are maintained. Then you see the 3 years grafted plants giving the economic fruits. Nucellus and grafted plant, nucellus and seedling produce fruits 10 to 12 years, but grafted plants give fruits, economic fruits 5 to 6 years. When we will be using the grafted plant, the farmers can be benefited at least 24 to 36 lakhs if we give the supply to the farmers, the grafted plant compared to the seedling. Now raising the nuclear citrus seedling, when I was in the Kalimpong, the tissue culture technique we have done because the twig of the mother plant we used for the explant for the development of seedling. Now, the problem of the rooting is there. Therefore, we have standardized the root tip, shoot tip grafting. Now, cross protection effort was made to produce the to identify the cross protecting milestone. Now, milestone means the milestone cross protection. When we will be inoculate the milestone, it can protect the super infections. And we have 21 isolates maintained in my glass house. And based on these things, based on this codon uses biasness, because when we have used the code-protein gene and suppressor genes of these 20 isolates, and we have sequenced and analyzed, then we have seen we have got two groups. That these two groups might be the predicted mild type, and others will be the severe type. And these all 1, 2, 3, 4, 5, 6. These 6 isolates may be the putative mild types. Now we have to challenge in the glass house level. Then we can identify the actual mild states. After that, biotechnological based management, we have made the gene construct, code-protein gene, the sense, antisense, and hairpin. Then we have standardized the transformation protocol with CTP genome construct, and we have seen the regenerated percentage is the 1.29, and we got PCR positive. What is coming this one? What is coming this one? A PCR positive plant we have also identified. After that, I could not continue, but again we have started after this bad situation. After that, now I am coming. The next disease, cotton leaf virus, and Dr. Pal Khurana is very much well known in these kinds of regards because I am always collaborating with him. Now it is known Indian the cotton position of the cotton in the Indian economy. That is the 12.43 million hectares cotton growing area India, and in northwest India it is 1.1 million hectares. But entire 1.1 million hectares of northwest India cotton growing areas are infected by the cotton leaf curl viruses. Now it is known that 1989 CLCUD has been reported. Professor Anupam Verma in 1989 in IRI field, and then 1993 Siganganagar epidemic was found in Sindh Pradesh, sudden flare up in Punjab and Haryana 1997. In 92 to 97, loss of $5 billion in Pakistan. Then 2008 to 9, resident varieties become susceptible. And 2011-12, the entire northwestern cotton growing area have been infected by the cotton leaf curl viruses. It is the white fly transmitted viruses, Gemini virus containing DNA A, associated with beta and alpha, producing the cotton leaf curl diseases. In India, is the total 5 cotton leaf curl viruses are present now. What we have studied during last 10 years, the incidence, then genetic diversity, and identification of virulent, dominant strain, recombinant strains, biotechnological based management. Now survey, we have made extensive survey all the cotton growing areas in the northwest India. After then we have estimated the disease incidence. You see the disease incidence is various in the different areas. The 37.5% incidence to the 12, then 13, then in this way, and the zigzag way. That now we have seen 3 epidemics: the 2001, 2002, 2006, and 2013. Now changing CLCUD virus pattern. You see the 2012, the Multan virus, the 2013 Multan virus, 2014 Multan virus, 2015 Multan virus, 2016 again Multan virus, 2017 Multan virus, and 2018 now Cochrane virus. That means changing of the pattern. When this now this is the situation 2015 to 2019, the disease is less because here all the viruses are recombinant. That's why they are they are going to fitting their pathogenicity. When they will be virulent, after that it may be coming another epidemics within very soon. Now total 21 complete genome of cotton leaf curl virus has been cloned, sequenced, and analyzed. And based on this, we have seen all the 21 isolates of India having 83 to 100% nucleotide identity among them. And when we have analyzed the phylogenetic tree, then we have seen the maximum viruses in India are cotton leaf curl Rajasthan virus, and very low number of the viruses are the Cochrane virus, that is Burewala virus. The present isolates, presently last 2019, we have seen again Rajasthan virus, and earlier also Rajasthan virus. But present Rajasthan virus are different from the previous Rajasthan virus in their sequence level. Therefore, the virus in this present condition, they are in the very dynamic positions, and they are now in the stable condition, and recombination is going on and going on. When some of the severe strains will be coming, and I don't hope the disease will be coming, so that the epidemic may be coming up soon. Therefore, we have to be careful. Now, recombination is the important criteria for the evolution of the strains in India. Now, I have studied the distribution, because in all the area, viruses are distributed. Four kinds of virus of the cotton leaf curl virus are distributed. Now, 19 beta satellites and 21 alpha satellites molecule have been cloned, sequenced. And India, the single beta satellite molecules are there, and 4 kinds of the alpha satellites also identified in India. Now alpha and beta satellites are also shown, they are very much recombinant. And now biotechnological waste management strategy I have effort has been made by me. Then I have made some construct of intern hairpin based on the BC1 gene and the port-protein gene. Then agrobacterium mediated implantar transformation. This is the growth of the transformant. Then we have tested the screening, antibiotic screening, then GRAS test, then PCR confirmation, that is RT-PCR confirmation. Now 49 overall plants we got last year, and this year we have got 75 plants. But achanak, I do not know. That project has been closed by ICR. I do not know what we will be doing within these T5 plants. Now conclusion. So Corona said, I need to say the conclusion. I will stop. Corona said, shall I continue conclusions? Now, shall I stop? The CTV incidents 16 to 70, overall 10 CTV variants in India, complete genome has been cloned, sequenced, and it is a declining inducing strain. Recombination are the major phenomena for evolution of CTV virus based on the 5 genome, one from Delhi, Bangalore, Katol, Vidarbha, Assam, the CTV of India, the distinct. The based on CP genes, CTV displays low codon uses biasness, higher genetic stability, mutation rate is very low. Citrus reticulata originated, CTV has higher codon uses. Citrus reticulata is an evolutionary primitive, and CTV might have been evolved millions years ago in Citrus reticulata, that is Mandarin. Produce CTV-free planting material, several gene construct has been made, develop citrus transformation protocol for production, identify putative milestones, and in cotton, the survey was made and disease was estimated from 2012 to 2019. 21 CTV virus complete genomes were characterized on RCA cloning, sequencing, sequence analysis. Majorities of them of the Rajasthan virus, followed by the Cochrane, Burewala, Faisalabad, and strain. Cotton leaf curl Rajasthan strain is predominant and distributed in most of the cotton growing areas of the world in India. 13 present Vigo virus are recombinants, where Rajasthan is the strong recombinant. The beta satellites and alpha satellites have been cloned and sequenced. All the present beta satellites are Multan satellites, and alpha satellites are the 4th species. Cotton leaf curl Burewala alpha satellites, cotton leaf curl Multan alpha satellite, Gossypium darwini symptomless alpha satellites, and cotton yellow vein mosaic alpha satellites. And complex interaction of recombinant cotton leaf curl Rajasthan, recombinant cotton leaf curl Multan beta satellite, and divergent alpha satellites are associated with the cotton leaf curl outbreak in India. And CLCUD derived gene constructs were made, and transgenic cottons to implantar transformation have been developed. Now, acknowledgement, Deputy Government of India, Director IRI, Professor Nupam Verma, Professor Y. Salawa, Professor Sen, Professor R.K. Jain, Dr. Ramachandran, B.K. Barnwal, B.G. Malti, and this is my power. Based on this power, I got such kinds of achievement. Then I am very much thankful and very much happy to acknowledge all the students who worked with me, Utpal, Vivek, Shruti, Supratik, Balaram, Shibu, then Shukla, Satish, many students with me for my research work. Thank you very much, and pranam.
Thank you, Professor Kajal. I'm really glad that you gave the marathon lecture in about 30 minutes, and impressive. Very good. I had not known some of the new things that you have done. Congratulations for that as well as the lecture award. And with this, I hand over the reins to Professor Manorachari so that Pramod can take over for his award lecture. Thank you very much.
Monorajari sir, unmute please.
कर दीजिए ना वहाँ ओके At the outset, I am thankful to IPS and the speaker, Dr. Biswas. He has touched upon many aspects, including the modern aspects like recombination technology and also complete genome sequences and control measures. And one important thing is that in this pandemic era, citrus has become very important plant, and the virus that he has taken, I think he will be able to produce a lot of healthy material for the farmers and also the cotton, and which is already known for the transgenics. And I congratulate him for getting this prestigious Dastur Memorial Lecture Award. And Dastu, in name of Dastu, there was one fungus which was named as Dasturella, which was an hyperparasite. Now, I would like to introduce the another speaker, that is Dr. Pramod Kumar, who is well known for his work in plant pathology. He is from JNKV Jabalpur and PhD in plant pathology. And he has been working on need-based technologies for the farming community. And more so, he is an extension plant pathologist and also very well known for his work among the farmers. And he has published around 40 research publications he has got to his credit. Very upcoming plant pathologist, so as to say. And he has received outstanding scientist award from that of Uttarakhand and best KVK scientist award. That is also very important one because whoever has done field work and helped the farmers, they will be getting this best KVK award. And he has received Young Scientist Award from SITE, Ranchi. And he has got many more feathers in his cap, MJ Narasimhan Award in 2011 and IPS Journal Counselor Merit Award, and because he was also counselor for this particular Central Zone, and Best Poster Award also he has received. And he has also received Best Oral Presentation Award in 2020, the international conference which was organized in Delhi. And he has been considered as a farmers-friendly scientist and a plant doctor. And I wish him good luck, and I congratulate him for getting this Sinha Memorial Lecture Award by the IPS. And IPS also needs to be congratulated for selecting two best upcoming scientists for this. He is going to speak on current states of cercosporide fungi and other aspects. May I request Dr. Pramod Kumar to join for presentation. Dr. Pramod Kumar. Dr. Pramod Kumar, please.
Sir, good morning, sir. Good morning, sir. Thank you. I want to play my recorded, I had sent the earlier recorded my presentations. So I request you to IPS to present my, play my presentation, sir.
Dr. Gogoi.
Hello. Hi, yes. So shall we share from here, Pramod?
Yeah, वही बोल रहे.
Okay.
Earlier 28th, sir, I have sent the recorded presentation.
Yes, yes.
Problem, you please upload.
Good morning to all of you.
आपके पास नहीं है तो फिर प्रमोद कुमार.
It is here. Just maybe.
Good morning to all of you. Today,
प्रमोद जी आपके पास है तो कर दो ना प्रमोद जी हेलो प्रमोद कुमार
Okay, sir, I will present here.
Some problem is there, Pramod, because file is not opening here. Hello, Dr. Pramod. Dr. Pramod, some problem is there with your file here. So you can share from your side.
Okay, I will try, sir.
Okay. Dr. Pramod?
Sir.
Does not come. I think you have to double click your file.
Sir, actually, some problem is there for the computer side. One of the is need the activation of the Office, Microsoft Office.
Because here when I'm trying to open it, it is automatically getting closed immediately. So some problem is there in your file, maybe issues with compatibility of the software.
Yes, PPT 2020. Sir, I need to open in another well, sir, just wait for 1 minute, sir. 1 minute.
Trying from here, just
1 minute, sir.
Maybe it's a too big file, that's why it is not able to do it.
Yes. When I'm trying, no.
Because 1.23 GB is such a big file.
Yeah, Babu.
Sir, I will. I need 1 minute, sir. 1 minute, boss.
ನಾನು ಲೊಕೇಶನ್ ಶೇರ್ ಮಾಡ್ಲಾ ಏನ್ ನಿನ್ ಬಾಬು ತಾನೆ ನೀನ್ ಸಾರ್ಗಿರ ಅಂತ ಅನ್ಬೇಡ ಬಾರ ನಿನ್ನ
Dr. Devappa, please mute yourselves.
ಲೊಕೇಶನ್ ಶೇರ್ ಮಾಡ್ತೀನಿ ನಂಬರ್ಗೆ ಬಾ ಬಾ
प्रमोद जी
Sir, just I will start, sir.
Yeah, good. Full screen कर दो.
Sir,
yeah, fine.
Audible, sir?
Yeah, yes.
Sir.
Go ahead.
Sir, good morning, sir. Good morning to all of you. I'm highly thankful to Indian Phytopathology Society for nominating me for S. Sinha Memorial Award, and I humbly accept the honor and dedicated myself to taxonomy of fungi as discussed by the Manora Chari sir. And today I have presented the current status of Cercospora fungi in India, effective management strategies, and future direction, which is need of present time, sir. Then you have to know Sir, the earlier recorded the fungi in Sarcospora fungi in Hedvidia during 1863. Just name is the Sarcospora Francis. In the all the systematic positions is there is belong to the Stomycota, the Ethereomycetides, Capnodius, and the Microsperaceae. They belong to Sarcospora Francis. And the type specimens have also is present in Sarcospora penicillin by Secardo during 1706, and the synonyms are the Virgosporium cookie having present in the Graviella. Then the are available in the. Any problem, sir?
No, it's all right. All right.
Okay.
No, go ahead.
Various literatures are available in the our country, like in Charles by 1954. Then 1963 by the R.S. Vasudeva, 1965 by Katsuki, M.B. Ellis, 1976, and Braun. And currently the Kamal 2010 enumerated the various Sarcospora fungi in India. And the Cross and Braun illustrated the 4 new Sarcospora fungi on the basis of their Conidia 4 and the Conidia size. This is the Sarcospora, Pseudo-Sarcospora, Passolora, and the Esternella. When we are going to the contributions of the Microspora family, you have to know the fungi are assembled in the Dematiaceous hypomycetes without microspora likes, and the sexual mark belonging to the Microsporolaceae, the genera Aspergillorium, Sarcospora, Detridospora, Passolora, Prathigda, Pseudo-Sarcospora. These are recognized as the Sarcosporids fungi, and they are the mostly cause the leaf spot diseases in any of the various flora of the plant throughout the more common in the human subtropical region, and are the mostly folliculous pathogens pathogenic to various host crops. And if you are going to for the distribution of the Sarcosporoid fungi, maximum contribution to Sarcospora fungi has been made from the Uttar Pradesh, followed by West Bengal, Maharashtra, Karnataka, Andhra Pradesh, as well as Telangana and Madhya Pradesh. These are when we are going to the kingdom fungi is important component of the biodiversity, and the mankind by UNEP 1992. The work with the biological diversity to the tropical fungi is immense. The total number of fungal species on the earth is estimated from empirical and theoretical status by Hawksworth during 1977. About 1.5 million of the which less than 8% are currently known. This number indicated as the estimated number of the fungal morpha or not, since most species have been, and as we'll describe on the phenotypic basis. If you are going to the classical approaches for the identification, they are some of the misleading due to the hybridizations of the pathogen and the cryptic speciations and the convergence evolutions of the fungi. Then the most is the humidity, that is the environment factor that is belonging to the is misleading due to the Sarcosporoid fungi. When we are studying about the classical approach of the identification of the Sarcosporoid fungi, the first is the name of the host and place of the collection is very important factor for the record of the fungi, and then the conidiogenous cells, conidiomata, and external mycelia is the other identifications for the Sarcosporoid fungi. And the conidiophore characteristics, pigmentation and ornamentation of conidia, and the thickness of the hilum, presence of expansions of the hilum, and the conidia safe incisors are the more approaches for the identification of the Sarcosporoid fungi. During I have associated with also with during 2005 and 15 we have the survey of the different area of the Madhya Pradesh, like Balaghat, Satna, total 16 districts we have visited, and the 21 Sarcosporoid fungi were recorded in medicinal plants and enumerated for the state as a new host record for the Madhya Pradesh. There are some photographs we have taken. I myself photographs taken during the 15 years. The Sarcospora on Blumia lesra and Japtor carcassa Pasolura Azrakai Blumia lesra Sarcospora pais Diplocyclos palmatus and the Erythrina subrus. These are the medicinal plants associated fungi, and this is the Pseudocercospora cochleae in Cocculus hirsutus diascoria species Ocimum officinalis of Abutula and Indicum. These are the host Jasmanium arborescens and the Lantana camera, Nectanthan arboretractis, Glamlia arborea, and Opercula terpethum. Pogostement franca plectran throids, Resinus communis, and the Tinospora cardifolia. This is the most important problem presently in the Azadirachta indica, Clitoria ternatica, Sarcospora ternatia, Vitis nugundo, and the Vithania somnifera, Ajasobanda, Elephantus escaper, Mentha arvensis. These are the hosts we have the work during the my 10 years data. When we have studied the Sarcosporoid fungi is based on the study based on the made Conidia and Conidia 4. Then the study is the cultural characteristics depend on the X type of the culture regulatory. The earlier the identification is very tough, but today the culture isolation of the culture is very easy. Then the better resolution we go for the molecular basis study. This is primary and secondary barcode of the Sarcosporoid fungi and the deposition of sequence in National Center for NCBI for the better resolution of the Sarcosporoid fungi. When we are going to the Sarcosporoid fungi, how to identify correctly? There are major things are the isolation direct from the host is very important for the characterizations in the morphology of the Sarcospora on the basis of we have earlier discussed the Conidia ornamentation, hilum, and the thickness of the Conidia 4 and the cultural identification. We have today a lot of culture oscillators the Sarcosporoid fungi. The primary and secondary barcode based analysis is very important for the identification, and the phylogenic analysis is the for the very important for the assessment of the species on the basis of morphology and sequence based study currently. When we are study with the problems with the identification of the Sarcosporoid fungi, the Sarcosporoid is not identified correctly due to the inadequate morphological characteristics to distinguish the species of the Sarcosporoid. The IPS is not able to distinguish the different species here today. The Sarcosporoid is not yet cultured today, not more. The culture media they grow very slow and often ends with the contamination is the major problem of the and the Sarcosporoid fungi is very high in spore productions, therefore difficult to prove the pathogenicity and host range if you are going to the Sarcosporoid fungi. The present requirement for the proper identifications of Sarcosporoid fungi, the India is considered 17th mega diverse country is the way to say survey of unexplored region is necessary for the foliar fungi, particularly the Sarcosporoid fungi, to understand the global biodiversity, and inoculations test is needs to confirm the taxonomic studies, and not a lot of number of crop fruit and forest plants have been infected by the Sarcosporoid fungi. And losses, if you have discussed with the losses, the 50 to 75% losses is caused by the Sarcosporoid fungi in our earth, and this reduce the quality of the fruits and vegetables, and the many secondary produce the many secondary metabolites, and for the correct identification, resistant source or genes in crop plants is very important, and the common fungicides can be recommended by the conducting the minimum field trials for the extensions workers in the country. If you are study about the current status of Sarcosporoid fungi of India, we have earlier discussed the Kamal 2000 compiled the total 1815 Sarcosporoid fungi approximately 1200 hosted from the different locations of the India, and based on the present study was observed that out of the species described so far, only 1304 included in microbank presently, and the 479 species of Sarcospora Cercospora included, and 476 species are Pseudocercospora, Passolora 149, and the Jasmarium 91, and the Esternilla 12, and the 12 species Sclerostigmata have been included in the. Only there were the 39 species spurious is reported in the various publications until 2018. We have seen, and out of the 1304 fungi, only culture, yet only 12 are available in national culture deposit like MTCC, ITC, and ARI Pune. Only 12 are available. If you study the current state of continuations, additionally there are other 9 species of Cercospora which are transferred to the 9 other Passolora calendly, and the Passola gentians as a Cercospora varicolosa, and Cercospora gentian, and the 470 species of Cercospora is recorded currently. If you are earlier discussed with the other our scientists, Cross and Braun 2003, they had described mainly on the structure of conodiophores, that is scaram hyla, and presence and absence of the pigmentation of conodiophores and the conodia, and the Cross during 2009 the study based on the phylogeny of the genus Micros, on the phylogeny of the Cercospora fungi on the morph of the. Then we are the molecular study started by the Gronewald in 2013 for the study secondary barcode genes for the species level identification is very important for the single university origin of the fungi Cercosporates. Then 2015 by Baxi confirmed most of the assumptions made by Cross and Boron relating to generic circumstances by the Baxi, and the suggested Cercospora genera into the separate class, that is the Palicellospora, Paracercospora, Phiocercospora, Porocercospora, and Neo Pseudocercospora on the basis of the Baxi 2015. If you have studied, then again Baxi 2000 studies the recommended on the encoding genes as the Calmodium encoding has recommended glyceraldehyde 3-phosphate dehydrogenase as a promoting gene for the species eliminations in the Cercospora by the Baxi during 2018. If you have studied the symptomatology and the histopathology of the Cercospora, spot occurs on all flora of the world. The disease is generally established only on the flores, but if the climate conditions are suitable, symptoms may be also come about. Appropriate use to the instrument. When we are going to identification and new records of the Cercospora fungi in Indian context, so various researchers, Sir Mororacharya is there, and Dr. Jamaluddin, Dr. Bill Gami is also various taxonomists is there. So researchers describe the numerous novel texts of hypomycetes of mycology and plant pathology of India, and on the basis of morphotaxonomic studies of these fungi were done through the routine method of microscopic examination, and with the help of prevailing literature and expertise available by Chapter during 1954, Cross and Brown 2010, and re-examined the Cross and re-examined the 3000 Cercospora on the basis of description, mainly on the arrangement of conodiophore loci, absence or presence of pigmentation, and conodia and conodiophore. If you are study the distribution and the host range of the fungi, Cercospora fungi is the single largest and most heterogeneous genus of the hypomycetes species of the Cercospora, distributes globally and cause leaf spot disease on the most of the major plant flora worldwide. Several species Cercospora described and illustrated from the diverse hosts and locations are morphologically impossible to differentiate, and cereals, pulses, vegetable, ornamental, other secondary weed host crop, including medicinal and aromatics plants and other forest flora, are also associated with the Cercospora fungi. When we are studying effect on the crop growth and economical losses, the average losses average losses due to this pathogen are estimated about 50 to 75 in crop plant. It reduces the quality in fruit and vegetables when the climatic conditions are not favorable for the development of infection. The disease may develop on the primary leaf apparatus foliage as the development of the infection, not entirely destroyed, nor regrowth occurs and damage or to probably the disease, the fungus survive between the infected plant residue crop growing seasons, predominantly in the form of stomata, structure that can persist for 2 years or more and account for the main source of primary income for the onset of the disease epidemic and initiation of the disease. When we are going to the dispersal and epidemiology of the Sarcospora fungi, the Protea of Sarcospora play a major role of primary inoculum in the disease incidence. Rain splashes are responsible for the disease of Protea. Sarcospora leaf spot is considered an important pathogen not only due to the widespread ranges but also due to the susceptibility of many commercial crops to the diseases. We study on the eco-friendly approaches for the management of disease. For the Protea of Sarcospora leaf spot of this is discussed the mung bean assess the different techniques and methodology included due to the chemical fungicides. The Sahazadi 2017 the effect less the disease incidence and the management practices of the crop rotations and use of non-host crop plant they destroyed by the less the disease incidence. Then we have study with the chemical Nath during 2013 use the tabiconazole one was found best to reduce the groundnut leaf intensity 52.4%, and then we have a biological control trichoderma based BOE based model where fungicide used by the leaf for the Kura and the Abdaru stack of the increase the body of the groundnut by the Hansen et al. Then we have discussed with the IDM practices. Jaipuria et al. 2011 had minimized the disease incidence of the Sarcospora leaf spot garden pea and capsicum. 11.18% was recorded by the treatment with the poultry manure, biofertilizer, integrated nutrient management, vermicompost, biofertilizer essence for Sarcospora leaf spot, capsicum, and garden pea. Then we have a study with the disease predictions model for the Sarcospora fungi. Various workers have been tilted. Currently, Bhardwaj in 2020s developed the prediction models for the grey leaf spot disease in fodder, sorghum, and the developed based on the 10-year data. It can occur during the 29th week of the grey leaf spots had very strong negative correlation by the Bhardwaj with the weak average temperature 0.7, while moderate correlation was found with weekly average relative humidity. When we are studying with the molecular diagnosis and genetic diversity of Sarcospora we have also reviewed the unpublished monograph on the Sarcospora fungi of India. It was found that there is little barcode information available in India. However, the additional genomic data can add in the better insights into the host pathogen infection and disease management. Total 935 species of Sarcospora reported in India by different researchers such as the Chap, Vastu, Kamal. Among these, Joshi in 2006 studied the genetic diversity of 11 different isolates of Sarcospora cancers, the causative agent of Sarcospora leaf spot in legume. This is the status of X-type Sarcospora fungi in India recorded in the National Fungal Culture Collection of India in Agarkar Research Institute, that is SE1, and MTC's microbial type culture collection in Chandigarh, 3 in our Indian type culture collection, that is 2. The total number of 12 Sarcospora fungi X-type culture have been deposited in our India. If you had discussed the genome sequence of Sarcospora causative agent of early leaf currently 1920 paper, Valerie and Orner have been deposited the sequence of Sarcospora erraticola, and 2015 by Orner the entire the NCBI gene bank database to the 8,070 base pair in the 21 sequence with the half of these entries. And the Satuta in 2019 reported the first drop genome of Sarcospora kupchi and the assembly of the pathogens. And currently 2020 in this our Kamal and the currently Sarma has associated with the Wilkin 2020 the drop the genome sequence of the Sarcospora citrulina and the brassicaceae. These are our current sciences are work with the sequence of the Sarcospora genome. Then the requirement for the publishing a fungal species, we have introduced that the descriptions must be appear in a general book form for publishing the Sarcosporous fungi or other fungi. Then the specific have been unique botanical nomenclature for the writing, and the new species must have the English and Latin diagnosis or description as we had with the taxonomical position. In the nomenclature data must be deposited in the approved repository like Mycovania index, fungus or fungal names, and the SSN number will be cited for the. And then the other is we are going for the molecular pendant, that the deposit DNA database in the repository such as the gene bank and the alignment of the tree base would be done for the current identification. We have discussed with the record available in the India monographs and our Indian type SCI have checklist of Sarcospora species are available in 2019. And the Kamal we have a report recently work on Sarcospora fungi of India, and the earlier the RS Vasudeva 1963 work on Indian Sarcospora fungi. These are the records available in Indian micro monographs in our India. Then we have discussed with the future directions for the advances of the Sarcospora fungi. The advances of any branch of science depend upon the methods used in the study and the essentials to continue the investigations on open method, which may be more useful, reliable, involving less expensive symbols, so that the simple say techniques may be usually conducted then reproduces work at the. When we are India is the considered earlier is the 17th mega diverse country in the world. Hence, a through survey of unexplored region is essential for the foliar fungi, particular Sarcospora fungi, in the order to understand the global biodiversity. And we have discussed in relations to the various components such as the methodology and relations to the cultural, chemical, biological management, and the applications of the classes of fungicide with the different modes of actions and the alterations and rotation of appropriate mixtures in combinations with the judicious timing of the applications is anticipated to successfully ensure their continuous expectation. One of the problems associated with the Sarcospora fungi, we have already get the promising result in similar approaches for the target fungi may enhance the capability of crop protections approaches in the future. If we are to face the universal threats into plant health and meet the challenges of the food security in a changing climate scenario, a more systemic and combined approaches is required for the amalgam matching plant breeding and biotechnology, chemistry, and rational policies on the sustainable use of pesticide, as well as continuing innovations in alternative crop protections technologies, the future action. Okay, thank you for hearing and listening. Thank you to Indian Fighter Panther Society, sir. Thank you, sir.
Well done.
Thank you very much, sir.
Professor Manohar, sir. I think he is. So, Professor Paul Kurana, sir.
Where is Professor Manorachari?
That's what I think he's disconnected again.
Okay. I think, the presentations, both of them were very good. The second one was equally exhaustive. The first one was really marathon-like. And I'm glad. I hope, now Pramod is at Jabalpur proper, not at Balaghat like before. And with this, I thank the IPS for inviting me, and also thank the secretary and all others, the ex and the present president, for the kind invitation and the arrangements. Thank you once again.
Thank you, sir. May I request, before that, we warmly welcome to our President, Dr. P.K. Chakraborty, sir, and also President-elect, Madam Pratibha Sharma, Madam, so to this session. And I think with this, may I request the chairs of this important session to virtually honor the awardee. I will share one slides. Just let me share the slide. So may I request now the chairs to honor our awardee with this virtual mode. So please join Paul Khurana sir and Manoj sir. Please confer and announce this to confer this award to observe the memorial award to Dr. Kajal Kumar Biswas. Paul Khurana sir, please unmute yourself and virtually confer.
Yes, sir. The Dastur Memorial Lecture Award is conferred on Dr. Biswas, and S. Sinha Memorial Lecture Award is conferred on Dr. Pramod Kumar. Both of them they deserve, and we appreciate the IPS for recognizing the talent of youngsters. And now lastly, I would like to conclude Dr. Pramod Kumar's lecture. He has worked on taxonomy, holotype concept, biodiversity, classical approaches, and as well as the complexities and problems faced in research in culturing, and then the molecular approaches and pathological aspects and disease management. I really appreciate his concern for the Sarcosporoid fungi, which is very complex genus, and it deserves all appreciation. Myself and Professor Paul Khurana sir, we would like to place on record our grateful thanks to the IPS, and also the presence of many of the scientists like Dr. Gogai and Dr. Mondal, Dr. Rashmi Agarwal, Dr. Sridhar, and Dr. Selvarajan, Dr. Devappa, Dr. Makesh Kumar is appreciated. We thank every one of you for having given us an opportunity to chair the session. We thank all of you. Thank you very much, sir. Thank you.
Thank you, sir. On behalf of the society and on behalf of the organizing committee, I extend my thanks to both the chairs for nicely conducting this awards lecture session. And I think it's a great privilege that we have both these stalwarts in plant pathology for nicely conducting this session. And we are also thankful to awardees, particularly Dr. Kajal Biswas, our beloved Kajalda, for J.F. Dastur Memorial Award, and Dr. Pramod for S. Sinha Memorial Award. So we are really grateful for your contributions, and your contributions has been remarked as for this award. So with this, I now request to switch over our next session, and it is a very important session for the APS Travel Grant Award Contest, and this session will be chaired by again two eminent personalities. So we stay. We are just because we are already past 10 minutes. So I think I may now request Dr. R.N. Pandey and Dr. A.K. Mishra to chair this important session. But before asking them to chair, just I may take the opportunity to introduce Professor A.K. Mishra as well as Professor R.N. Pandey to the August gathering. So Professor R.N. Pandey, Ram Narayan Pandey worked at different capacities at the Department of Plant Pathology, BA College of Agriculture, Anand Agriculture University, Gujarat. And he started his career from assistant professor, and then he took charge from professor and head of the Department of Plant Pathology. He is credited with the publications, a substantial number of publications, 5 books, 8 book chapters, and 45 research papers. He remained the principal investigator of different projects, and he also credited with the establishment of a state-of-art laboratories, molecular characterizations of plant pathogens, and trichotoma productions. His major interest remains detection of plant pathogens, pathogenic variability, genetic of host parasite interactions, epidemiology, and chemical and biological management of diseases, and recommended 28 technologies for the disease management to the farmers. He worked in at different capacities to the societies also, and he remained president in the year 2018, as well as zonal president 2010, and he has been awarded with different from different societies, and he also our honored fellow of the IPS. So, and also awarded with different other society. So with this, I welcome Professor Pandey to chair this session. Along with, I also welcome Professor A.K. Mishra to chair this award ceremony, and to brief about this Professor A.K. Mishra, who is well known to all of us, a recipient of award by President of India, was project coordinator of AICRP on subtropical fruits and head of the divisions of crop protections from Lucknow. His outstanding contributions is well known for in the field of plant pathology and horticulture, and he is credited with a very, you can say, large number of publications and in a reputed journals, and 572 publications and 88 books, bulletins, and proceedings. Professor Mishra is recipient of award from President that includes Rajiv Gandhi Rashtriya Gyan Vigyan Mallik Pustak Lekhan Puruskar by Home Ministry, Lifetime Achievement Award by DAHDS organizations, and Dr. Punjab Singh Distinguished Scientist Award by UPCAR, Sardar Lele Memorial Award in the year 2017, K.C. Mehta and Manoranjan Mitra Award, and Plant Pathology Leadership Award in the year 2013. He also, we are happy that he also served the society as a president of IPS, and under his leadership, we also move ahead. So with this, I again request Professor A.K. Mishra to chair this session. With this, I request both the chairs to start the proceedings. And before starting the proceedings, it is just to mention to the speakers that APS Grant Award is each from each zone. They have been nominated, and we have to recommend all of them to the APS Grant Award. But again, we have to prioritize them, and for this, that's why this is the contest. So thank you very much, Professor Mishra and Professor Pandey.
Okay, thank you, Dr. Mandal, for introducing to the gatherings. And I'm grateful to the IPS to providing me the opportunity to host, to chair such important session of APS travel grant of the student which have been recommended from different zones. So respected Dr. P.K. Chakravarti, present president of IPS, Dr. Mandal, then other participant and present over here, my colleague and friend, Dr. Mishra ji, Dr. Rashmi ji, Dr. Pratibha, Dr. Mahesh Patil ji, and all other who are very much familiar to us in this IPS family. So in this, I welcome you all to this very important session. And as told by the Dr. Mandal that the time allotted, there are 5 students who will be contesting for this APS travel grant. And I will name, and those who are present, they kindly tell. Mr. Jagmohan Singh is from Division of Plant Pathology, I see the IARI, New Delhi. Then second is the Gautam Kumar Rautu, Assam Agricultural University. Shumia Wani from Sere Kashmir University. Then Miss Sabia Saeed from Department of Plant Protection, Aligarh Muslim University. Vaibhav Pratap Singh is also from Aligarh Muslim University. So these are 5 contestants from different zones who will be presenting their work to us. And the time allotted to each of them is 15 minutes. And so with this, I invite my co-chair, Dr. Mishra ji, to say something if you want.
Thank you, Dr. Pandey. First of all, I would like to thank Indian Phytopathological Society for chairing this session, which is very important. I remember that this APS travel grant started during the period of international conference when I was president. And it is a good step, and all the contestants I welcome, and I think that it is very useful activity, and I wish them very success. And I will not delay more. I will just like Dr. Professor Pandey to start the proceeding with the presenters. Thank you.
Yeah, thank you. Thank you, Dr. Saab. And I'm just told by the secretary, Dr. Gogoi, that there is one presentation left yesterday. That Savarni Tripathi from IRA Regional Station, she will be presenting her work, her lecture, that is oral lecture. So if she is here, she can present her lecture.
So Savarni Tripathi sir abhi join nahi kiya because he is not connected.
Okay.
Just let me see whether Savarni.
Yeah, sure.
Or otherwise, you can start with Mohan Singh.
I'm here,
Hello,
sir.
Raju.
I think he is present.
Yes, sir. Yes, I am here, sir.
Okay.
We should not waste time.
Oh, sure.
Okay. So should I present, sir? Or?
Just 1 minute. Dr. Mandal? Yeah, the first minute, please.
Sir, 1 minute. Hello. So, sir, Dr. Sabani is already joined. So,
okay.
Dr. Sabani, are you prepared for presentations right now?
Yeah.
Oh, then it's fine. So, with the permission of the chair, sir, may I request then Dr. Sabani to share the slide?
Yes.
Yeah. Please finish within 15 minutes, okay? Dr. Tripathi.
Okay, sir.
Thank you. Please share your slide.
Can you see my slides?
Yeah, sure.
Yes,
Yeah, it is there.
Slide mode. Please put in slide mode.
Slide mode. Yeah. It's okay. Okay, please go ahead.
Okay. So first of all, I would like to thank the IPS and the chairs to allow me to present in this session because yesterday there was network issue, so I could not able to present. And so in this today's presentation, I'm going to briefly update about the current situation of the viral diseases infecting vegetable crops in western part of Maharashtra, and also highlight some of the effort we are making to develop the management modules with special reference to tomato and chili. As we know, Maharashtra is one of the leading states of vegetable production in the country, and this soil-mixed vegetable, especially tomato, chili, and capsicum, are important for the country's nutritional security and also for the better return to the farmers. So, indeed, the viral diseases are a serious concern for vegetable cultivation, as it appears from the pictures you can see in the slide, and also evident by the news articles appears in media time to time highlighting these problems. Because the virus infection which severely affect the yield and market quality of produce, most of the time farmers are not able to get the invested return. So many times it seems that the crop are either abandoned or produce is wasted as they don't want to add additional inputs and cost for transportation and marketing it. And there is another serious concern of the heavy and indiscriminate use of the pesticides and other agrochemicals, which adding the higher cost of cultivation and damaging the ecosystem, and also raising concern to the nutritional security and health issues. As you can see in the picture, this is a real picture taken from the farmer fields. And this indiscriminate use of agrochemicals play a key role in developing resistance to the insect vectors and also makes them hyperactive, and thus their efficacy of virus spread is noted higher. So keeping in view of the above concern, we have initiated a survey to know the virus disease situation in this region. The main focus area was western part of Maharashtra, and which has intensive cultivation of vegetables. We covered around 180 villages and more than 600 acres of vegetable field over several years. And we found is that indeed viruses are major limiting factor for vegetable cultivation and is affect the market quality, yield and economic return. It is also noted that the viral diseases showing increasing trends over the year with possibilities of new emerging and re-emerging viruses. The introduction of new varieties and germplasm by seed industries, indiscriminate use of agrochemicals, and changing climate might be acting as enhancer for developing serious disease and pest problem in the region. Many times, farmers experience very heavy losses, and sometimes is noted more than 100%, almost like no return, and sometimes even they don't get invested inputs. Here are some of the examples showing extent of damage caused by the virus complex. The upper panel of the picture of capsicum cultivation under protected cultivation, set up near to Kolhapur districts. Almost 100% plant are severely affected by the virus complex with almost no fruit production. Even if few fruits are there, they have no market value. The same situation is also observed in case of open cultivated tomato near Narayangaon area, which is one of the largest tomato producing belt of Maharashtra. Another example is chili crop, which was devastated by virus disease complex in several districts around the Nagpur region 5 years back. And this problem was severely affected the chili production and also impacted the export of red chili to the Middle Eastern countries. This is to just give you an idea about the different type of viruses infecting vegetable crops in this region. This is clearly shows that the several viruses associated with vegetable crops, and most of the time there are more than one viruses associated with particular crops. So it's become very complex to manage them. Moreover, the maximum number of viruses are associated with solanaceous vegetables such as tomato, capsicum, and chili, and they were shown to have more than 10 different virus infections, and most of the time more than 2 viruses are recorded to associated with the complex. Overall, it also shows that most of the viruses belongs to Otivirus, Cucumovirus, Gemini virus, and Tospovirus group, and mainly they are transmitted by thrips, whitefly, and aphids. Now, this is established that the virus are serious concern to the vegetable cultivation. So we started looking the strategy to manage these diseases in the integrated manner. The major consideration of developing the management module was that it should be practical, means that is easy to adopt by farmer and easy to use. Second, it should be effective in minimizing the losses caused by the virus complex and maximizing the quality, yield, and economic return. And the third one is very important, that management practices should be safer to environment and ecological friendly. Because there is no chemical available to directly control the viruses and no resistant variety available for viral disease complex, especially in case of vegetable crops. So the option available is to modify the cultural practices and the vector control. So we started designing the virus management strategies by testing different components which might be effective to reduce the losses caused by the viruses. The key component tested were different pesticides, biopesticides in separate or in combination to control the virus spreading insect vectors and modifying the cultivation practices such as use of different color plastic mulches and organic mulches and application of floating row cover in separate or in combination, that best combination we utilize. Several experiments were conducted to show the effectiveness of these components on virus and quality yield separately and in different combination. A variety of selected permutation and combination were tried to get the best results and to develop a management package or module for a specific crop with a specific location. Another important component in management strategy was used the border and barrier cropping, and this was also effective in managing virus problem with other combination in certain situations. This is in picture, you can see the 3 rows of pigeon pea with maize, which was used in managing the tomato diseases in Kharif seasons. Okay, this is the pictures as mentioned the previous different combination of treatments. In earlier slides, the several components and different modification practices were tested separately and also in combination over the years. Finally, we come with some cultural modification practices which are effective and easy to use in present situation and found to be significant in mitigating the virus complex problem in some of the vegetable crops suitable for this region, especially the western part of Maharashtra. This is one of the example of management module we developed for tomato. In this management trial, you can see the slide, the use of silver mulch, crop cover, and these were in combination with minimum application of insecticides. The visual of lower panel clearly provide an idea about yield and quality produced in the treatment as compared to the control. If we look at the disease, situation that's shown in upper left side graph, the use of silver mulch with crop cover and spray of that is called Beneria at 2-week interval after removal of crop cover found to be the best in reducing the disease. And this is just whole treatment is only 2 spray. This shows less than 1% disease incidence after 75 days in best module, whereas control has 7 times higher disease incidence. Similarly, the vector population was also monitored at 45 and 75 days, shown in lower left-hand side, which also indicate the lowest population of aphid thrips and whitefly. The outcome of best treatment is also reflected in terms of marketable yield, which is important for the farmers, as shown in the right-hand side graph. The best treatment showed almost double the yield as compared to the control. And the similar situation was also recorded in case of chili trial with the treatment with silver mulch, crop cover, and insecticide. And here we have some adjustment having the insecticide plus biocide application, that is agroneem based. Some more is spread because the virus disease is very high in this situation. Okay, if you look at the data, the result of chili trial shown very similar pattern as tomato trial. The disease incidence in best performing treatment, that is silver mulch, crop cover, and minimum spray of insecticide and combination with the agroneem, has reduced the incidence up to 20% after 75 days, whereas the control it was close to 100%.
Dr. Tripathi, 3 minutes only, please.
Okay, sir. Okay, this treatment combination was also found to be the best in minimizing the insect vector population, as you can see in the pictures. And if you check at the yield, also is more than 2.5-fold higher as compared to control. Okay, so with that, the management module was found promising in managing the virus disease complex in our research farm. So we wanted to try at the farmer field. Therefore, the we tried at the farmer field in Narayangaon area, where is a major tomato growing belt. If you see the data of the performance of the management module, which consists of silver mulch crop cover and 2 spray of Vinivia, has been very promising and consistent in enhancing the yield and disease incidence for 3 years, as it can be seen in the slide. The similar situation also tried in case of chili, and that was, as you can see in the farmer field, we conducted the trial at the real farmer field where we had hot spot of the disease. Here if you see that we wanted to look at the upper right side of the graph, it shows that the high disease incidence is close to 80% in the normal farmer practice, while significant reduction of the disease was recorded in the developed module where we have less than 30%. And to develop the management module was also increasing the yield of about 1.4 fold as compared to the farmers' normal practices. Based on the above validation data, we therefore feel that the absence of suitable and market acceptable resistant varieties, this management module is effective option in managing virus disease complex in vegetable crop in this western region of the Maharashtra. I hope this module will be helpful to the farmers in other region. We have to try this practice to other region with minor adjustment, and I hope it will work, and hope this will add some value to the vegetable farmers. With that, thank you very much.
Thank you, Dr. Tripathi, for a nice presentation. Now I invite Mr. Jagmohan Singh. Dr. Mishra ji.
If anybody
yes, please.
having any, can I make one request to ask one question?
Yeah. Only one question, please, because we will be running out of time.
So, Savarni Tripathi ji, it is a very good presentation, comprehensive on the management of viral complex in Narayangaon. I understand you have made a very good areas of different management practices. Number one is that did you also calculated the economics of which is the best cost effective to understand, because a lot of farmers are small holding farmers in Narayangaon and Nasik area. Perhaps they also look for economics of it, and also did you consider any resistant variety to include in this kind of management practices?
Okay, the first question, that economics part of it, that we are still calculating the economics part, but roughly I can give you an idea that how it works. Because we calculated in that area, like depend on the situation and the disease prevalence. But the farmer can gain up to 60 to 1 lakh rupees per hectare using this module. So it's depending on the situation. Okay, the second thing that there is no resistant variety we could see in the market so far for this virus complex. So we do not go for the resistant, we go for the popular variety the farmer are using.
Okay, thank you. Maybe I'll come by email or phone.
Sure. You can communicate each other for your problems. So thank you, Dr. Tripathi and Dr. Suresh for live discussions. And now I invite the participant, contestant of the IPS travel grant to present this one. And in this, the first speaker or presenter is Mr. Jagmohan Singh from IARI. So best of luck to Mr. Jagmohan ji. Please share your slide and go ahead. The time allotted is 15 minutes. After 12 minutes, you will get a warning and you have to summarize within 3 minutes. Okay.
Thank you, sir.
So I request Dr. Mishra ji to please monitor the time.
Hello.
Yeah.
I welcome you all to my presentation for IPS Travel Grant. My title for presentation is Deciphering the Catamine Globosum and Induced Signaling Networks in Tomato Against Early Blight. If we look for the plant disease management, most common method is chemical control, but it is environment hazardous, so we have to look for the alternative options. Alternative option is biocontrol agent, and most common used biocontrol agent is trichoderma. So here I'm introducing you the Cotomium bulbosum. Recently commercialized biocontrol agent in market fungus belong.
Some sound problem is there. Can you rectify?
It was first reported by cuisine in 1870. More than 300 species of Cotomium are known. Only the Cotomium bulbosum is
Because
most
it is
commonly
recorded one, Dr.
isolated
Sahab.
and used as
So I think
biocontrol
as
agent.
he is present, so he can present also directly. That will be.
If you
Hello,
look for
Yeah.
Jagman, you can also present because there is some
the
sound problem.
morphology of this pathogen turns later dark in color, short neck,
Please, you can
and
present directly.
on its
Dr. Jagmohan, are you prepared to present orally?
short on.
He is not listening, it seems.
Yeah.
Dr. Jagmohan?
Dr. Jagman.
Hello.
Jagman.
Yes, sir. Yes, sir.
So
Can you present orally?
Yes, sir.
Because
Okay.
sound problem is there.
Yes.
Yeah.
Yes, sir.
From
I'm audible, sir.
from next slide. Yeah. Yeah. From
next slide, no
you are
problem.
audible.
Yes.
From next slide. Yeah. Not from beginning, but after that, the last slide which you have presented.
Slide mode.
Yes, sir.
It in slide mode.
Sir,
Yes,
if I
slide
put in
mode,
slide
please.
mode, then voice is coming. So I have
Acha,
to-
okay, anyway, please go ahead.
You can clear the voice, sir, so that
You can clear the voice from slide. Just go to the that speaker symbol, and there you can mute also. Or from the slideshow also you can mute. You can erase all the encoded sound encoding.
If you look for the morphology of this pathogen or this fungus, it is cottony white in color, which turns later brown to black in color. It produces a fruiting body called
Yeah, that's
perithecia, which
good.
is dark.
Good.
Yes, sir.
Yeah, yeah. Now
If you
you
look
can
for the
present
morphology
orally,
of this
please.
pathogen or this fungus,
I
It's
think
not
it's
been
coming.
clear, I think.
No, it is cleared, I think.
Just see.
Yeah, yeah. Now
Yes.
it is cleared. Yeah.
Yes.
Yes, sir.
Yeah. Please go ahead after that.
Yes, sir. Coming to morphology of this fungus, this is Cotomium bulbosum. It is cottony white in color, which turns later red to brown in color and even black in color. It produces a fruiting body called perithecia, and which has a large number of setae coiled around it. It produces the ascospores of lemon in shape and brown in color. And if you look for the antagonistic effect of this fungus, Cotomium bulbosum is a phytopathogenic, is effective against various plant pathogens like bipolar serotonin, solani. And again, if you look for the biocontrol mechanism, we have basically the antibiosis, mycoparasitism, induced resistance, and competition. A number of reports are available about the antibiosis and mycoparasitism, but a little reports are available on induced resistance. So our focus was to study the induced resistance component of Cotomium bulbosum. For this induced resistance component, the increase in the expression induced resistance increase in the expression of natural defense mechanisms of the plant in defense mechanism of the plant by range of factors. Like there are two type of induced resistance, SAR and ISR. SAR is systemic acquired resistance, which is correlated with increase, which involves the salicylic acid pathway and the activation of PR genes. And ISR is activated by non-pathogenic rhizospheric fungus and bacteria, which involve the jasmonic and ethylene pathways. Coming to the research gap, this fungus biocontrol mechanism of induced resistance is not well studied. So no information is available about the different pathways and crosstalk between the pathways. And it's better to study the induced defense component so that it could be manipulated to enhance the plant resistance. And for this, the title of the research was deciphering the Cotomium bulbosum induced resistance, induced signaling networks in tomato against early blight disease. First objective was to evaluate the effect of Cotomium bulbosum induced defense on the severity of early blight disease, and second was the transcriptome profiling of tomato for deciphering the induced defense pathways. Then validation of transcriptomic data by monitoring the expression of upregulated genes of different signaling pathways. For the valuation, to evaluate the effect of chromium bulbosum, alternary chromium bulbosum on the induced on the severity of early blight, plants were inoculated, pre-inoculated with chromium bulbosum and counter-inoculated with alternary solani, and disease scoring was done at different time points. You can visualize the symptoms produced by alternary solani, and if we for scoring the disease, we calculated the percentage disease index, area under disease progress curve, and rate of disease infection. There was significant reduction in the percentage disease index up to 30 to 35%, and we evaluated the effect on the plant growth and development. We got that there's a significant increase in the plant height and root length, and also the photosynthesis. Other physiological parameters like photosynthesis and the stomatal conductance, transpiration, these were also upregulated. The plant growth parameter like root length and shoot length increased by 20% and 59% respectively. And the other physiological parameter like photosynthesis, transpiration, and stomatal conductance enhanced by 54%, 145, and 101%. And for the transcriptional profiling of tomato, the plants were inoculated with CZ at 3 leaf stage, and samples were collected at different time points. RNA was extracted and cDNA was synthesized. The cDNA was taken for the RNA for sequencing by using the Illumina high-seq platform. Raw reads were collected. Those raw reads were trimmed first, then high-quality reads were taken to generate the de novo assembly, and then de novo assembly transcripts were transdecoded. By using transdecoder, CDS were predicted. Those CDS that coding sequences were further differentiated into differentially expressed genes on the DSEQ2. Then these were functionally annotated by using BLAST2Go, and these DECs were further classified into different KEGG pathways using the CAS. And these are the DEGs. Total 22,000 DEGs were present, and we compared with various combinations: 0R to 12R, 0 to 24R, 96 hours, and 12, 24, 24, 96. And we observed 922 DEGs at 12 hours and respectively in others. When we compare the expression of these differentially expressed genes at different time points, and first is 0R to 12R, we can see the 80 genes which are significantly expressed more than twofold change at 0 hours, 61 at 61 genes only at 12 hours and 781 at both the time points. And similarly for the 0 to 24 and 0 to 96 hours. And these are the heat maps, and heat maps display the upregulated and downregulated genes. And these are the most of the genes which belong to phenylpropanoids pathway or related to signaling pathways. And these were further Go analysis was done for this DEGs at 12 hours, and which represent the three categories to which these DEGs belong: belonging to biological processes, molecular function, and cellular component. If you look into biological process, most of the DEGs belong to cellular process, metabolic process, response to stimulus, or regulated response to biological response, and signaling. And overall, we looked into the Go analysis of this transcriptomic reveals that most of disease has catalytic activity, binding activity, and metabolic process belong to metabolic process, response to stimulus, and cellular component. And further, the top 10 KEGG pathways were observed, and these is most belonging to the top 10 pathways at different time points are represented here. And most of these belong to the metabolic pathway or biosynthesis of secondary metabolites and protein and plant hormones related, plant signaling related hormones. And if we look into the specifically into the signaling process of the plant, defense signaling, it is carried by the different phytohormones like salicylic acid mediated, monic acid mediated, and brassinosteroid mediated, and ethylene mediated. Salicylic acid mediated is related to SAR, and other two are related to ISR. And we observed that the genes which are red in color are upregulated and green in color are downregulated. At initial stage at 0 to 12 hour, all the genes present at the initially signaling like ETR and JAR, NPR, these are expressed. And at 24 hours, these are at the same rate, and the signal is carried further at JAG or TCH at the end of this. And similarly, at 96 hours, earlier genes are downregulated, and the genes present at end of the pathways are highly upregulated. This shows the sequential expression in the gene and involvement of jasmonic acid and signaling acid pathway for signal transduction. When we looked for the phenylpropionate biosynthesis pathway, and we observed that the genes, most of the enzymes like PAL, and these are highly upregulated. You can visualize the red in color. These show the upregulation of these enzymes of phenylpropionate biosynthesis pathway, which further lead to synthesis of salicylic acid and various other secondary metabolites. And as we compare 0 to 12, this for the 12 to 24 hour, the genes which were earlier upregulated, those are downregulated, and it is few other genes are upregulated. And at 96 hours, all the genes above in the pathways are downregulated, and it is there's sequential expression in this pathway, and the genes which are present at the bottom at the end of this pathway are upregulated at 96 hours. The most of enzyme involved in the phenylpropionate biosynthesis pathway are upregulated in sequential manner from upstream to downstream, starting from 12-hour post-inoculation to 96-hour post-inoculation with CG. And if we look for the enrichment of phytohormones, as I show the earlier results, there was increase in the growth parameters and various physiological parameters. And so we observed then we screened this for growth-related hormones like auxin, cytokinin, gibberellin, and abscisic acid. And most of the genes present in cytokinin are upregulated, and whereas the gibberellic acid pathway remains as such, it is this is not expressed. And at 24 hours, we can visualize the expression of most of the genes related to auxin, and sequential progression in cytokinin signaling and abscisic acid. And here there's at 96 hours, there is or at all the time points, the gibberellic acid pathway is not expressed, and cytokinin pathway is at the same pace as earlier at 96 hours. And plant growth and development related plant hormones like cytokinin signaling are activated on inoculation with CG to tomato plants. And further transcriptomic data was validated by using various markers like markers for jasmonic acid pathway, salicylic acid pathway, and ethylene pathway by using QRT-PCR. In similar process as mentioned earlier, RNA was extracted and cDNA was synthesized, and that was used for real-time PCR by using cybergreen, and data was analyzed by using delta CT method. And we selected for transcriptome validation, we selected the genes which are belonging to different defense signaling pathways. Like first, you can observe the first 4 genes, LOX, JAR, and PIMC. These are related to the jasmonic acid pathway. Then PR1, PAL, PYL, PLD were key factors like 31, 11, 17 related to salicylic acid pathway. Then some related to ethylene pathway, another related to phenylpropionate pathway, or some peroxidase, chitinase, and respiratory burst related to another defense-related genes. And there was, if we observe the genes, these all related to salicylic acid pathway, PR1, PAL, PYL, and PLD. And there is significant increase in is a significant fold increase is observed, and mostly at 12 hours post inoculation, and up to 10 folds or 8 folds. And this was in correlation to what we observed in transcriptomic data. Similarly, the worky transcription factors which are activated downstream in signaling salicylic acid signaling pathways, these are also upregulated at 12 hours, 24 hours. And genes related to jasmonic acid pathway like LOX, JAR1, these are upregulated at 6 hour, 12 hour, and upregulation is highly upregulated as compared to salicylic acid pathway up to 20-fold or 25-folds. And this was very similar to what we observed in transcriptomic data. And these are the genes which are related to ethylene pathway. These are
Dr. Jagmohan, you have to conclude.
Yes, sir. And these are upregulated, but to a smaller extent, only up to the 2-fold. And these are the genes related to the another defense pathways like peroxidase and respiratory burst. And overall, it is that we observed that CG is involved in CG inoculated plants, salicylic acid and jasmonic acid pathway is upregulated, and it indicates the involvement of ISR and SAR. Coming to the summary, systemic induced defense by CG in tomato decreased the early blight disease by 30 to 35%. And whereas there's upregulation in, there was significant increase in
Vishwaji,
root.
please take care. I will take 2 minutes.
There is significant
Vishwaji,
increase
please
in
take care, okay?
After CG inoculation tomato, there's significant increase in growth parameters and other physiological parameters like photosynthesis, transpiration, and stomatal conductance. And transcriptomic data reveals the 900 diseases at 12 hours and respectively at 24 and 96 hours. The KEGG pathway analysis indicate that diseases mostly belong to the secondary metabolites or the plant hormone signaling transduction pathways. And GO analysis revealed that most of the diseases belong to the catalytic activity, binding activity, response to stimulus, and defense response. The genes related to plant defense such as peroxidase, chitinase, or rapid oxidative burst are highly upregulated at on inoculation with the CG2, and which is revealed by transcriptomic data and qPCR. Jasmonic acid and salicylic acid signaling pathways are highly upregulated in tomato at different time points after inoculation with the CG2, and whereas ET signaling is upregulated only to limited extent. And upregulation of jasmonic and salicylic acid pathway indicate the involvement of ISR and SAR in mechanism of induced defense by CG in tomato. And thank you one and all for patience listening. Thank you.
Thank you, Dr. Jagmohan Singh, for excellent presentation, and you rightly deserve this award. I will request the Secretary IPS for the next presentation. Perhaps it is Gautam Kumar Gothu, whether she available?
Sir, yes, sir.
Please.
Am I audible, sir?
Yes,
Yeah,
yes.
you are audible, yeah.
Present your slide.
Share your screen, please.
Full screen you can make. Nice.
Yes, sir. Are my slides visible, sir?
Please go ahead.
Yes, sir. So, good morning, respected dignitaries. Myself, R. Gautam Kumar, PhD student from the Department of Plant Pathology, Assam Agriculture University, would like to present on the topic post-transcriptional gene silencing using code protein gene-specific double-stranded RNA molecules against cognate cucumber mosaic virus infecting Bujulokia crop of Assam. So, in this research, we made an attempt to inhibit the replication mechanism of cucumber mosaic virus in Bujulokia. Bujulokia is considered as one of the hottest chili in the world, is having a wide economic importance. So the crop is being attacked by a wide range of viruses, out of which the cucumber mosaic virus is considered one of the major viruses. So with the help of RNA silencing mechanism, we made an attempt to inhibit the virus infection in plants. So these are the contents of my research. Coming to the introduction, every year a huge economic losses has been reported due to the plant viruses diseases, and also plant viruses are highly diverse and use a variety of strategies for their multiplication and spread. That is why it is very difficult for the control of the viruses in plants. Viruses are also vectored by a great variety of invertebrates. Thus, intensive chemical control is not an option for the control of the viruses. Public is demanding for ecological control measures for crop protection has created a need for alternative and chemical control. So in this regard, RNA silencing mechanism provides a better scope for the virus control. Coming to the introduction of the crop, the king chili, the Bujulokia, is considered as the hottest chili, is extensively grown in Assam and northeastern India. The cucumber mosaic viruses is considered as one of the most primitive viruses and causing a huge economic loss to this crop. The virus is known for the well-devastating nature and causes economic loss of more than 60% in the field. And the CMV comes under the genus of cucumovirus under the family of the Bromaviridae and is transmitted by the aphids by a non-persistent manner. Here we can see the symptoms of the cucumber mosaic viruses on the Bujulokia. These are the reports of the cucumber mosaic viruses where we have reported the incidence of cucumber mosaic virus and prevalence in Assam and northeastern India, where the plants exhibiting the severe curling and exhibiting mosaic patterns and typical filiform symptoms on the leaf. These are the photographs which we have observed that the typical symptoms of cucumber mosaic virus, where the filiform leaves, where the leaves has become very thin in structure and also mosaic pattern on leaves. Coming to the genome of cucumber mosaic virus, the genome is consisted of single-stranded RNA and consists of RNA segments of 1, 2, 3, and 4. and each gene is specific for its own function. RNA1 and RNA2 encodes for the proteins which are involved in genome replication and internal transcription, whereas ORF2b encodes for silencing suppressor gene which suppresses the gene silencing mechanism, whereas RNA3 encodes for movement protein for the movement of one cell to other cell, whereas the RNA4 encodes for coat protein gene which encapsulates in viral encapsulation and cell to cell movement. So in this we have selected the coat protein gene of cucumber mosaic virus. So if we develop a double-strand RNA construct against this coat protein gene, it will be able to regulate the uncoating of the virus and reduce the spread of virus in plants. Coming to the mechanism of RNA gene silencing mechanism, it was initially proposed by Jorgensen in petunia plants. Later, the Craig and Mello has developed the technique for which they have received the Nobel Prize in 1998. Double-stranded gene silencing mechanism is a sequence-specific process which specifically targets the mRNA and causes degradation and causes the protein block and ultimately leads to the silencing of the gene. Gene silencing is initiated by a double-strand RNA molecule. Whenever a double-strand RNA molecule is introduced into the cell, a dicer-like protein cleaves these double-strand RNAs into short interfering RNAs. These short interfering RNAs moves into the RISC complex, that is known as RNA-inducing silencing complex. Argonate protein plays a key role and cleaves the siRNAs into single strands, where the guide strand moves to the translation machinery and blocks the gene. And ultimately, the protein synthesis is blocked and the gene is silenced. So taking this as a background, we have constructed double-strand RNA and we have introduced into this cell. So here we have constructed double-strand RNA of gene of interest and we have introduced this into the plant cell, and actually the process of gene silencing will take place. Taking this as a background, we have drawn the objectives as development of double-strand RNA construct for cucumber mosaic virus resistance in Burgelokia. So after constructing the double-strand RNA construct, we have performed a proof of concept studies for CMV silencing in Burgelokia using double-strand RNA as a strategy. So coming to material methods under the objective 1 for the construction of double-strand RNA, total RNA was extracted from the suspected leaves by the Trizol method and by the kit method. This is the protocol followed for the RNA extraction by Trizol method. And through the kit method, we have also isolated the total RNA from the suspected leaves. So after isolation of the total RNA, we have estimated by quantitative and qualitative. So quantitative estimation is done through spectrophotometer, whereas the qualitative is done through gel electrophoresis. So this is the gel picture that was observed after the qualitative estimation of total RNA isolated from the leaves. So after isolating the total RNA, we have designed the primers. So for the target gene to amplify, we have designed the primers by using the primer 3 software. Here we can see the forward and reverse primer with a product size of 593 base pairs and with a melting temperature of 53 degrees centigrade. So once the primer were designed, we have checked the efficiency of the primers that are able to produce the effective siRNAs once they are introduced in the cell by keeping a sequence in siRNA software. So here we can see the effective number of siRNAs that the target region that they can produce once they are introduced into the cell. So after the primers we have designed, we have run the reverse transcription PCR, that is conversion of RNA into DNA, by using the protocol by adding the gene specific primers. So after conducting a reverse transcription PCR, we have loaded the samples into gel electrophoresis. Here the picture of the loading into gel electrophoresis. Once the samples became positive, we have eluted the gel and also purified the gene of interest. So this is the picture that is obtained through the amplification of the gene. Here we can see the 593 base pairs product of a gene of interest, that is coat protein gene of cucumber mosaic viruses, was observed, and this gene was eluted and purified. Once this purified product was, we have cloned the gene of interest into PTZ vector, that is the cloning process was started. So in the cloning process, the first step is the ligation, where we have ligated our gene of interest into this vector by following a standard protocol. So this is the protocol followed for the ligation. After the ligation, we have transformed this gene of interest into this DH5 alpha competent cells. So here is the protocol that is followed for the preparation of competent cells. So for the preparation of competent cells, we have used DH5 alpha strain. For the poration to occur, we have used calcium chloride. So this is the transformation protocol followed for the transformation of our gene of interest into competent cells. So after process of transformation, we have spread those colonies onto albe agar plates and have kept at 37 degrees overnight. So here is the picture that was observed on the next day, where we can see the transformed colonies were observed on the LB agar plate. So here we have to confirm whether our gene of interest was placed in the correct orientation are placed in the vector or not. So for the analysis to prove, we have performed the restriction digestion analysis to confirm whether our gene of interest was placed in the vector or not. So for that restriction digestion analysis, we have isolated the plasmids from the recombinant colonies. So this is the protocol that is observed, that is followed for the isolation of plasmids. Once the plasmids have isolated, here we have performed the restriction digestion analysis by using the restriction enzymes, the ECOR1 and BAMH1. This is the protocol followed for the restriction enzymes. So ECOR1 and BAMH1 are added, and after the restriction digestion analysis, we have loaded the samples in the gel electrophoresis. So here is the restriction digestion analysis gel photograph. Here, the upper band, we can see that the vector of 2.9 kb and our gene of interest was placed at correct orientation, that is 593 base pairs. So after the confirming of restriction digestion analysis, we have gone for sequencing. So after sequencing, we have obtained the results. Here we have, after getting the sequencing results, we have blasted those results in the NCBI website. Here we can see that our isolated cucumber mosaic virus sharing a homology of 97.95% with the other sequences in the world. Hence, it is confirming that isolated sequence with the cucumber mosaic virus. So after sending the results to NCBI, we have got the accession number and we have drawn the phylogenetic analysis. Here is the phylogenetic analysis of the isolated CMV of Jorhat, sharing the homology with the Teok isolate of Assam. So once confirming all the results of sequencing and the restriction deletion, the CMV code protein product was again subcloned into a other vector that is known as L4440 vector. So this is the protocol followed for the subcloning into L4440 vector for the production of double-stranded RNA. The ligation and transformation was performed as per the protocol, and also we have analyzed the recombinant clones by restriction digestion analysis. So here the restriction digestion analysis were performed by BGL2 and XH1 restriction enzymes. So here is the gel picture that is observed by the restriction of L4440 vector and our gene of interest. So here we can see the L4440 vector of 2.5 kb and our gene of interest is a 593 base pairs. Here it is confirmed that the gene was perfectly oriented into this vector. So after this, we have proceeded for the production of double-stranded RNA. So here we have placed our gene of interest in between these T7 promoters of L4440 vector. So this vector is expressed by induction of IPTG, that is known as isopropyl thiogalactosidase. So once IPTG added to this vector, the vector will become expressed and the double-strand RNA is produced. So this is the protocol followed for the double-strand RNA production. So here we have induced this IPTG. So here our gene of interest, and we have induced this with IPTG, and upon induction, this double-strand DNA is transformed into double-strand RNA. So once the double-strand RNA is performed, we have pelleted out the cells and we have diluted this double-strand RNA with RNase-free water. So we have obtained the double-strand RNA and we have loaded into the gel electrophoresis. Here is the picture of double-strand RNA. Here we can see the gel picture indicating this is the double-strand RNA where it is induced with IPTG, where in this it was not induced with IPTG, that's why it is not producing any double-strand RNA. So after confirming the double-strand RNA was successfully produced, we have further moved to next objective, that is the assay test. So for the assay test to perform, we have raised the Burjolokia seedlings in a controlled conditions. So here is the picture of Burjolokia seedlings. So the seedlings were performed after the test for 20 to 30 days after their emergence. So we have tested in 3 treatments. That is the treatment 1, where we only with CMV viruses without any DSRN treatment, that is known as non-vaccinated plants. Whereas in treatment 2, we have inoculated the DSRNA along with CMV. So that is we have noted as vaccinated plants. And with the treatment 3 as control with only water, we have inoculated. So this is the protocol followed for the inoculation. So for the mechanical injury to occur on the leaves, we have dusted the silite powder on the leaves. So after the injury has occurred, we have dusted or sprayed the double-strand RNA on the leaves of Burjolokia. So here we can see the image of spraying of a double-strand RNA on the leaves of the Burjolokia plants at 20 to 30 years old as seedlings. So after the 25 days of post-application, here we can see the clearly demarcation of difference between the vaccinated plants and non-vaccinated plants. Here it is the picture of non-vaccinated plants showing a typical virus symptoms, whereas in vaccinated plants there is no virus typical symptoms and producing a virus growth, no virus growth, indicating that these vaccinated plants are free from virus infection. Here it is indicating that this double-strand RNA assay strategy could be employed in a defense against these viruses. So these are the readings that was taken. So in CMV inoculated plants, that is virus inoculated plants, has shown the reduced leaf size, whereas with dsRNA treated plants, that is vaccinated plants, have shown with normal leaf growth. So here we have performed the experiment with the two replications. So in the two replication, we have observed the similar results. So in the plant height also, we have observed that CMV inoculated plants have shown the reduced height, whereas dsRNA treated plants have shown the more height, indicating that thus dsRNA could interfere with viral RNA replication. So to further confirm whether any viral inoculum load is present in between the vaccinated plants and non-vaccinated plants, for that we have collected the leaves from both the treatments, that is from the CMV inoculated plants and also CMV dsRNA inoculated plants. So here is the picture, gel picture, that is observed at 35 days of post application. So in CMV inoculated plants, the viral load was clearly indicated, hence they are generating amplicons of 593 base pairs, where in dsRNA treated plants, no such viral inoculum has been present, indicating that these plants are free from viral infection. Here it is a clear demarcation that this dsRNA could interfere with the virus replication and cause to enhance the plant growth without any virus. Coming to the discussion, according to the Tenlado and 2001, they have reported that the direct application of this dsRNA onto the plant tissue at the virus inoculation confers resistance against the homologous viruses. And also Kaldas et al. In 2018 reported that the exogenous application of these double-strand RNA molecules could show resistance to the tobacco mosaic viruses. And also in Konakalla et al. In 2016 reported that the external application of dsRNA could confer resistance against tobacco mosaic viruses. So these are the summary and conclusion of my whole over the research. The code protein was successfully identified, and sequencing results has analyzed that they have shown the 97% similarity with other sequences of the world. And also this CP gene was successfully subcloned into this L440 vector. And also the subcloning was confirmed by the restriction digestion using BGL2 and XH41 restriction enzymes. So after the successful subcloning, we have further proceeded for the double-strand RNA production. So this double-strand RNA production was successfully performed by using protocol developed by Volodakis et al. So when this double-strand is produced, we have applied this double-strand RNA on the leaves of Bujulokia along with CMV. So we have seen that the viral replication was reduced. So the vaccination strategy could further be tested against different plant viruses infecting economically important crops, where virus occurs and cause considerable crop losses. So this RNA-based vaccination approach reported here could enhance the effectiveness of plant protection schemes for chili plants in nurseries and fields against CMV. Coming to the future prospects, so these dsRNA vaccines could be formulated as a dsRNA pesticides using nanotechnology, so that it could be directly applied in field as a pesticide spray against plant viruses. So with all this, I conclude. Thank you.
Thank you. Thank you, Doctor. You have completed in right time and excellent presentation. And Professor Pandey will be commenting something.
Nice work and nice presentation. So thank you, Dr. Gautam. Now next is Sumaya Vani. Are you ready?
Yes, sir, I'm ready.
Yeah, please share your slides.
Sir, is that?
No, not coming.
Not coming.
Share your slides.
Sir, I'm sharing.
No, sure.
First open and then share.
First you open on this desktop.
Slide mode, you turn to.
Yeah. Okay. It's coming. You are audible also, please go ahead. Wish you good luck, best of luck. Yeah, please start.
What happened?
She's logged out. I think she has got again sound encoded, so maybe required to delete the sound encoding first. Okay. So Vani, Sumaya Vani,
No, she's logged in, logged out, I think. Hello. Shumia Bani, are you hearing us?
Yes, sir.
Yeah, please share your
Sir, is my screen visible to all of you?
not now.
No, not now, not now. Earlier it was visible, but
You just share your screen. This is coming, so you just click it on and share your slides.
Sir, I am sharing, but I don't know what is happening.
No, initially it
and then
was
share.
sound encoded. So
You are sharing sound encoded earlier one or your own? Yeah, now it
Sir,
is
I have
coming.
both. I have both sound encoded and.
No, don't put sound encoded.
You
just
present it personally.
now it is visible. Now it is visible. Coming.
It is coming. Please now speak.
Now we can speak. Sumia.
Hello.
It seems there is some problem. Dr. Mandal, you can.
I think you can request the next one so that we can manage the time. Yes. Chairman sir.
Yeah, sure. Now Miss Sabiha Saeed, are you ready? Next contestant.
Miss Sabiha ready? Saeed.
Sabiha Saeed, are you ready?
Yes.
You present.
In the meantime, if you are just sharing one, I request Miss Sabiha Saeed to present.
Okay.
Yeah, please share your screen.
Avia, are you sharing your screen?
Yes, sir, I'm doing.
Yeah, please.
Again,
Shumia is coming.
This is again Wani.
Sir, is my presentation visible this time?
Yeah. It is present, but you go to slide mode.
Okay, sir.
It is coming on the screen. Now just click on to the slide mode, so you will be able to present.
All presenters should be ready with their.
Sure.
At least open the respective files so that. Yes.
Is this visible this time?
Yeah.
Okay, I'm really sorry for the inconvenience. I don't know what was happening.
No problem.
Anyway,
Okay.
you just start.
Okay, I'm highly thankful for providing me this opportunity. My name is Maya Vani, and I have done my PhD in plant pathology, and currently I am doing my PhD. Okay. I don't know, sir, what is happening. I'm not able to change this.
Next slide.
No, next. Yeah.
Okay. I welcome you all to this presentation. Okay. As all of us know that potato is the staple food and highly recommended security crop that can help shield the low-income countries from the international food prices. Likewise, in Kashmir, the potato is considered as one of the major crops. And likewise, other crops, it is susceptible to various fungal, bacterial, and viral diseases. And if we talk about the viral diseases, potato virus Y is the major virus that is affecting potato throughout the world. That results in economic loss of 80 to 100%. So keeping in consideration the importance of potato crop, the economic loss caused by the virus, and limited availability of studies in Kashmir regarding PVY, this research entitled Status and Molecular Characterization of Potato Virus Y Strains in Kashmir was planned with following objectives. Our first objective was survey to document the present status of PVY in Kashmir. And second objective was identification and molecular characterization of potato virus Y using universal code protein and strain-specific primers. And our research started with surveys at different selected locations and recording incidents at different selected locations, collection of samples, isolation of RNA, and then synthesis of CDNA from the isolated RNA. And later on, for the identification of virus, code protein specific primers were used, and for identification of different strains of this virus, strain specific primers were used. And later on, the representative samples from each location, they were sequenced, they were sent for the sequencing directly as PCR product, but after purification. And then also the retrieved sequences that were retrieved from the sequencing, they were blasted in NCBI for identification of our carrier sequences. Also, the phylogenetic analysis of the sequences was done, and little bit of study on PVY vectors in different potato fields was also carried out. Now, coming to first objective, that is survey to document the present status of PVY in Kashmir. As I already told that different locations were selected, we selected different districts of Kashmir, and from each district, equal number of locations were selected and almost equal number of samples were collected from each location. Here in this map, I have given the locations, different locations and different districts of Kashmir that we have targeted. And after I have of districts from where we collected the field. But I want to make this point clear here that we have targeted mainly the commercial potato fields of the Kashmir, as well as samples were collected from the local fields, but main target of the study was to target the commercial potato growing areas of the Kashmir. And during sample collection, samples were collected on the basis of peculiar symptoms of the virus, that is mosaic patterns on the leaves, stunted growth, puckering, leaf formation. And in addition to these peculiar symptoms, the peculiar sign that is presence of vectors on the, that is aphids on the infected samples was also taken into consideration. Okay. Sample collection was done in a random method. That's random method of sampling was followed. But mere collection of samples from any part of field doesn't mean that it actually represents our field. Here in this diagram, I have given the rough representation that how sample collection was done. Samples were collected from all the four corners as well as from the middle portion of the field, and almost 3 leaves per plant were collected. Properly, they were labeled, they were stored in liquid nitrogen and brought to the lab conditions. Okay, here in this table, I'm giving the incidence of PVY on the basis of symptomatology. But as all of us know that symptoms are not the actual, they doesn't give us the actual presence of the or actual identification of the virus. As virus symptoms, they always get confused, coincided with mineral deficiencies or other biotic and abiotic stresses. So this incidence is just mere representation of, this gives us mere idea whether the virus is present or not. Okay, coming to second objective, that is identification and molecular characterization of potato virus by using universal and strain-specific primers. Here I am giving this systematic representation of PVY genome. Actually, to give this diagram, the objective of this diagram is that the genome of potato virus is a single-stranded RNA virus, and it forms a matchword polyprotein that is cleaved into 10 matchword proteins later on. So for the identification of the virus, we have first targeted this code protein region has been targeted for the identification of virus. Primers were designed and code protein gene was targeted. And these are the gel pictures first showing the RNA isolated that I have already said that first of all we isolated the RNA from the collected samples. These are the gel pictures of the RNA, and later on using code protein specific primers in the PCR, these are the bands showing the presence of the virus in different isolates of PVY. Okay, later on we again calculated the incidence of the virus. This is the actual incidence that we calculated upon the basis of code protein specific results that we got. And in this incidence, it was seen that maximum incidence was found in Srinagar area of the Kashmir Valley, that is 52.38%, and minimum incidence was found in Badgam area, that is 25%. And later on, for the identification of different strains, different strain specific primers were used. Here I am giving the band size at straw of those primers, but these primers were already designed by Lawrence et al. That we have used it in our study. Okay, and this table here, I'm showing the distribution of different PVY strains in the districts of Kashmir Valley. And one important point to note here that in almost all the districts, mixed infection of the virus was noted. That is, mixed infection of different strains was present in almost all the areas. Here I am showing in the gel pics the different strains, mixed infection of different strains. That's NTN strain having the band size of 452 base pair. This is N strain, and here it is NTN strain and N strain. This is O strain having the band size of 267 base pairs. That means this is giving us the evidence. This is giving us the proof of mixed infections in different samples. Later on, the specific bands, representative bands from different locations, they were resolved on the gel. They were eluted using gel extraction kit, and the purified PCR product was sent for the custom sequencing. And these are the sequences that we obtained. We submitted them in NCBI, and we used them for our further study. This is the electropherogram showing the sharpness of peaks representing the exactness of nucleotides, likewise for other stamps. Later on, the phylogenetic analysis was carried out and for further confirmation, and our isolates were, these are our isolates from Kashmir F8 NTN, SL3 NTN, and phylogenetic analysis was carried out for further confirmation of study. And this gives us the proof that our isolators actually, they belong to different PVY strains. Later on, a bit of study was also carried out on the aphid vectors. We collected the aphids from the infected samples. We directly crushed them using liquid nitrogen. Likewise, for other samples, as I already told, we isolated RNA. For aphid samples, we also extracted RNA from the aphid samples. We synthesized cDNA and used a different code protein and strain-specific primers for the identification of virus in the aphid samples. But we were able to report only one strain, that is NTN strain, from the affected samples. Okay. We also did screening of available germplasm, that is the varieties which were available in the university. Also different varieties were collected through the from the commercial potato growing areas, from the farmers field. And during off season, these varieties were grown under the greenhouse conditions, protected conditions, and it was seen that most of them were showing the infections, showing the symptoms of different viruses. And in addition to PVY, we used the samples for PVX, PVS, PLRV, and it was seen that in addition to infection of PVY, these samples were showing the infection of PVX and other viruses in our seed potato. As we know that we in Kashmir, mostly we use the seed again and again that we store. Local farmers are mostly use the seeds of their own for the next season. So this virus gets proliferated again and again. So this is one of the major concern which we have to look into. So my study, we conclude the study and gave its future perspectives. This study gave us the brief insight about the prevalence and spread of PVY infection in commercial potato fields of the Kashmir. And the availability of specific primers will facilitate the typing of PVY infection at various isolators to the strain level. And the study laid the ground for the further investigation to determine the extent of PVY infection in particular, and possibility of other strains and other viruses in seed and commercial potato fields. And breeding of potato cultivars resistant to PVY infection should take into consideration the full gamut of PVY strains. Thank you so much. And I'm once again very sorry for the inconvenience.
Thank you very much, Vani, for an excellent presentation. And you rightly deserve this.
I'm really sorry, sir. I don't know what was happening. I'm
It's all
really
right.
sorry.
It happens. It happens sometimes.
Yeah, it happens. No problem. Please.
Dr. Pandey.
Yeah. It is good. Very good. Nice presentation. Thank you very much.
Thank you.
So
sir.
we'll go to the next presenter, Miss Shabiha. Miss Shabiha,
Yes.
are you there? Yeah, please share your slides and speak.
So Ani, you just unshare, and then Savia, you share the slide.
Miss Savia, are you there?
I'm sharing.
You unmute your speaker, please, and also on your video.
Please start,
Put
Saiba.
on your video and also your this one. Unmute your speaker. Yeah, it's okay. Now also share your video.
Presentation. By the time next speaker should also be ready,
Yeah, sure.
Vaibhav Pratap Singh.
Savia, is there any problem in sharing?
Yes, sir, there's some problem. I'm sorry for this.
So, did you submit here your slide?
Submitted.
Just let me check.
Dr. Manda. Meanwhile, can we go for the next presentation? Because time.
Sure, sure.
Mr. Singh, are you ready? Vaibhav Pratap.
Vaibhav Pratap Singh. Whether he is here?
Vaibhav Pratap, are you there?
I have not seen him earlier. I just.
Okay, no problem. Then Savia, please share. I'm not seeing you on the screen. Your video is also not on.
It seems there is some problem.
Savia, you can reply.
Yes, sir.
Yeah, you can put on your video also. Video is also closed. Yeah, now it's coming.
Not coming.
So you just open your PowerPoint, which you have prepared, and then share on the screen, please. Ms. Shavia, are you listening us?
Sir, I'm doing.
Yeah.
What is the problem? You should be ready.
Have you opened your presentation?
Yes, sir.
You open on your
Yeah.
laptop or whatever desktop, and then share. First, you open the slides.
Okay.
PPT. Yeah, open the PPT.
Yes, sir.
Dr.
Mandal, you are having a slide?
No, sir. Actually, in this folder which I got, that is there is not. She is not there. So maybe I have asked office here, that IPS.
No, she will be doing. She will be doing.
So,
have you opened your PowerPoint?
Yes, sir. I opened it.
Yeah.
And share it.
Now you click on the share screen, then it will come. Is there someone to help you?
No, sir.
You are alone?
what is the problem you are facing? Just.
Yeah. You just tell your problem,
It's also
please.
not on. So, Sabia.
Yes, sir.
Because we did not find your slide here. So I have asked again to recheck from the folder. But in the meantime, you try.
Wait, sir.
Could you open your PowerPoint?
Yes, it's open, sir, but I'm not able to share it.
You just first now you just there is a share screen.
Share screen.
You just click over there, share screen. It will come then automatically.
You have
but
to
your PowerPoint should be opened and on your screen, your laptop screen. First, you open your PowerPoint on laptop screen. Then share the screen. Right now, what problem you are getting? Please tell.
Actually, I'm clicking on share screen, but nothing.
Have you opened your PowerPoint on the screen of your computer?
Yes, sir.
Your laptop or computer?
Yes, sir. It's open.
It's open now. Then click
It's
on
open.
over this share screen.
Yes, sir.
Are you in the department? Nobody is there to help you?
No, I'm not in the department.
Okay. So no one is there to help you. Please try your best.
Well, Dr. Mandal, any alternative for that?
Sir, I am contacting. Maybe we got just now. Just let me check. Yes, she has sent late, so that's why it is different folder. So I am sharing from here.
Okay, okay.
Okay, share please. Yeah. Miss Shabia, why we could not see you on the computer screen here, over here, in the list of participants? Your
I'm here, sir.
photograph is not coming.
Yes, sir. We got it.
Audio
I'm
is not.
sharing from here.
Audio is not, video is
Audio is
not
there,
on.
video is not working. It seems.
I'm sharing from here, so we got it.
Okay, please.
Sure, sure. Yeah, it's good.
Slide mode you can.
Yeah, now Miss Shavia, you speak, please.
Just start within. Within 10 minutes you should finish because we have lost time.
No problem. She said we given 15 minutes. You have 15 minutes to your disposal, Miss Shavia. Don't worry.
Already the sound encoded. She started.
Sound is very low.
Very low.
Code.
Mm.
So
You can increase the volume.
Whether she will present.
As it is from sound encoded, I'll see just.
She will personally present or.
But sound is very low. We are not able to
listen.
Hear it. Yeah, listen it.
Just let me try.
Yeah, again. Miss Savia, are you able to share?
Can you improve the sound?
Yeah, improve. You can increase the volume of your computer. You just check the volume of your computer, Dr. Mandal.
Yes, sir. Achha, there it cannot be increased, no? Achha, yeah. Achha. Yes, yes, yes. Got it. Huh? This one. So then again, you have to share it. Yes, sir.
Yeah.
Everyone, I'm Sabir Saeed, and my presentation topic is the efficacy of some fighter attacks against moving elements India virus in Urpin.
Again, very low.
Sir, I have put it maximum.
But it is not clear.
But Miss Savia, are you there?
Yeah, I can explain.
You can present directly.
You can present, and in the meantime, Dr. Mandal, what you do, you just clear all the sounds from the slide.
Okay.
Yes.
Just a minute.
Just 1 minute, please. You just open this one and clear all the narration from all the slide. Clear narration from all the slides. There will be two options will be coming, timing and narration. So from all the slide, you have to remove the narrations. Dr. Mandal, is it over?
It seems there is some problem.
Are you able to do, Dr. Mandal? Is also off the screen.
Sir, I'm trying. It is almost done. So
Okay. So yeah, please take your own time. Yeah, thank you.
पानी पीते रहिए डॉक्टर साहब.
बस हो गया सर.
हाँ तो वही चलेगा.
जो डालता है अच्छा.
नहीं, it is not. अच्छा इसमें आ गया.
ये क्या है? कुछ इको आ रहा है.
नहीं आ रहा. तो maybe.
Shabia, your PowerPoint is not working?
No, sir.
हाँ, उसको सबका वॉइस स्लो करना पड़ेगा ना फिर. हाँ, वो भी तो समस्या है. Actually, एक का तो sufficient नहीं है, but it is done already. I think it is done. These only 2 slides are remaining.
No, you just open recording and there you just clear all the narration from all the slide.
Sir, that command is not actually operating. So
one by one you are.
So I have done it, but yes, fine. So I am sharing it now.
Sure. Thank you. Yeah, good. First slide. First slide. Ms. Shabia, are you ready now?
Yes, I'm ready, sir.
Yeah, go from the first slide and please take hurriedly and adjust your time, but patiently, don't worry. Present coolly.
Yes,
okay?
sir. Good
Okay. Good luck to you.
From the Department of Plant Protection, and I'm going to present on the topic bioefficacy of some phytoextract against mung bean yellow mosaic India virus in urd bean. Among several legumes, urd bean, also known as black gram, is an economically important drought-resistant pulse crop. It is mainly grown in Kharif. As urd bean is a leguminous crop, it improves soil fertility by fixing the atmospheric nitrogen in the soil. It is highly rich in protein, potassium, calcium, iron, niacin, thiamine, and riboflavin. It is widely cultivated in many tropical and subtropical regions of the world, including India, Pakistan, Bangladesh, Nepal, Myanmar, Malaysia, Iran, East Africa, and many southern European countries. In India, it is mainly cultivated in Uttar Pradesh, Madhya Pradesh, Delhi, Punjab, Bihar, West Bengal, Maharashtra, Andhra Pradesh, and Karnataka. Urd bean suffers many
Please synchronize your slide. Your slide is coming number 1, but you are speaking on another slide. So go on third slide, please. Dr. Mandal, please go on third slide.
Some issues are there.
Third slide, yeah.
Slides are not moving, sir. Yes.
Yeah, now next one.
Next.
That's all. Please continue, Ms. Savia.
You should say next, then he will change it.
You just say next, Ms. Savia.
Okay. Virus causing yellow mosaic disease is one of the most destructive diseases of urd bean, and it causes significant yield loss. It was first reported by Narayani in 1960. Yellow mosaic disease is transmitted by vector Bemisia tabaci. Symptoms first appear as small yellow flecks in young leaves. Subsequently, emerging leaves exhibit more conspicuous and irregular yellow and green patches. Yellow mosaic disease causes yield loss up to 10 to 100% depending on the stage at which crop is infested. Virus belongs to the genus Begomovirus of the family Geminiviridae. In urd bean, 3 species of Begomovirus are known to cause yellow mosaic disease, and these are mung bean yellow mosaic India virus, mung bean yellow mosaic virus, and horse gram yellow mosaic virus. And symptoms are almost same, caused by all these 3 species. Several approaches are there for the management of this disease. Use of chemical insecticides reduce disease incidence very efficiently, but these chemicals are very hazardous to the environment and cause harmful effect to the human health. Several non-host crops contain antiviral and insect repellent properties. Antiviral properties of the plant extracts in controlling viral diseases have
No.
in
Previous
controlling
one, Dr. Mandal.
Actually, it
have
is
Previous
in
one.
earlier
sir,
report
it is actually getting
plants.
automatic because sound though removed, but it was removed. I think from individual.
Achha, timing also you remove.
Yes.
You have also given the timing. Now clear the timings from all the slides option.
So I have to go there. Yes.
Yeah, again, please. Timings and narration both are coming on the slide. So if you remove the timings also.
Sir, you tell the next speaker also that they should be prepared.
Yeah, but Dr. Vaibhav is there, but he's not telling. I think he's not there.
He's not there. Okay.
Yeah, I think. Mr. Vaibhav Pratap Singh, are you there to present? He's not answering.
Anyway.
No problem. She's the last one.
It is not
Okay.
coming here. So anyway, we are going to.
Yeah, to this only, and just remove the timings. And go to the Miss Savya, which slide you are speaking? Which slide you are speaking? Third, fourth? Yeah, previous one, please. Yeah, this one. Yeah, please speak now.
Plant extracts are biodegradable, non-toxic, easily absorbed by the plants and do not leave any residual effects. In the present study, an attempt have been made to manage yellow mosaic disease of oat bean in eco-friendly way. Next.
Yeah, good. Yeah, please speak.
Yes. Diagnosis of virus involved in causing yellow mosaic disease in oat bean. For this, samples were collected from the experimental field and processed at IPR Kanpur. For the identification of virus, polymerase chain reaction was assayed using primers specific for coat protein gene of mung bean yellow mosaic India virus, mung bean yellow mosaic virus, and horse gram yellow mosaic virus. Annealing temperature was previous one.
Previous one.
Yes.
Yeah. Go ahead, please.
Annealing temperature was 56 degrees centigrade.
Yeah, please. Go ahead.
Next.
Hello. Next.
Now, after identification of the virus, full genome, next step was full genome characterization. For this, we designed 4 sets of primers, 2 for DNA A for the identification of 2 DNA A and 2 for DNA B. And the reaction mixture of 25 microliter contains a DNA template, 1 microliter primer forward and reverse primer, 1 microliter each. Dream Tag Green Master Mix 12.5 microliter and water 9.5. Annealing temperature was 56 degrees centigrade. Overlap amplicon size which we get was overlapped fragments of different size. Amplified product was sequenced and analyzed further to get the full genome. Next slide.
Back.
Eco-friendly management of yellow mosaic disease.
Yeah. Yeah. It said come now. Please speak. Yeah.
The experiment was conducted at the Faculty of Agricultural Sciences, AMU, Aligarh, and the treatment was laid in randomized complete block design. 2 sprays at 20 and 45 days after sowing were applied. Next.
Next, Dr. Mandal. Yeah.
Preparation of
Yeah.
For the preparation of phytoextracts, 500 grams of each Allium cepa, Azadirachta indica, Datura, Stramonium, ginger, garlic was taken, and it was washed 3 times, and then it is crushed in pre-chilled mortar and pestle, and then strained. And after straining, it was centrifuged at 15 degrees pre-chilled. At 15 degrees, 15,000 RPM per minute. and then the supernatant was collected, and these supernatant was then diluted further for the use. Next.
Coolly, don't worry. Yeah.
And these are the 16 treatments which were applied in the experiment. Next. Observations which were taken, data were recorded from 15 days after sowing at 15 days interval. Disease incidence, percent disease control, and disease severity were calculated by data recorded at 15 days of sowing using following formula. Yellow mosaic disease severity was calculated using 0 to 9 scale. Data were analyzed statistically using R software. Next, please.
Yeah.
These are the different parameters which have been taken in the experiment, and these are white fly count, disease
Back. Previous one, Dr. Mandal.
nodules per plant, shoot length, yield, kg per hectare, total chlorophyll content, rate of photosynthesis, and rate of transpiration. Next. Now results, gel photographs of the first experiment, and this band showing, and these are the band shows amplification with the primary specific for mung bean yellow mosaic India virus, confirming the presence of mung bean yellow mosaic India virus associated with the yellow mosaic disease. The bands is of 1000 base pair. Lane M shows DNA ladder of 1 kb. Lane 1, 2, 3, 4, 5, 7, 9, and 11. It shows the infected sample, and lane 6, 8, and 10 and 12 shows healthy samples. No amplification was seen when we have amplified using the primers specific for horse gram yellow mosaic virus and mung bean yellow mosaic virus. Next, please.
Next, Dr. Mandal. Yeah.
Virus identity. In the
Yeah. Virus identity slide, please.
Earlier one.
Yes.
That's.
In the present experiment, mung bean yellow mosaic India virus was identified as the cause of yellow mosaic disease using non-primers in the PCR. DNA A fragment showed 98.14% nucleotide identity with the DNA A molecule of mung bean yellow mosaic India of existing database of gene bank. DNA B fragment showed 95.81% identity with DNA B molecules of mung bean yellow mosaic India virus of existing database of gene bank. Next. Effect of phytoextracts on whitefly count. Treatment T3, that is Datura stramonium, at 20% was found most effective in lowering whitefly count after both spray. Whitefly was counted next day after spray. Next, please. Effect of phytoextract on mung bean yellow mosaic India virus incidence. It can be seen that among all the treatments, the Datura stramonium was found the highly effective, and all the phytoextract was found effective in controlling the disease, in lowering the incidence. But the most effective one was Datura stramonium at 20% concentration. It controls the disease by 48.71% compared to control. Next, please. Previous one. Effect of phytoextract in the suppression of yellow mosaic disease. Plant extracts contain antiviral substances that interferes with virus multiplication, resulting in the suppression of the disease. Graph shows that all the treatments helps in the suppression of the disease severity compared to control. Maximum suppression and minimum disease severity was recorded in treatment T3, that is Datura stramonium, at 20% concentration. Next, please. Figure 1 shows that the shows the plants infected with mung bean yellow mosaic India virus, and figure 2 shows the plants treated with Datura stramonium. Next, impact of phytoextracts on growth parameters of urd bean. Plant extracts were recorded effective not only in controlling disease, but also found fruitful in boosting several growth parameters. It was found effective in increasing pods per plant, nodules per plant, and shoot length. Maximum number of pods per plant, that is 37, and root nodules per plant, 32.6, was recorded in treatment T3, that is Datura stramonium at 20% concentration. However, treatment T12, that is an alien SIPA at 20% concentration, showed best in increasing shoot length compared to control. Impact of phytoextract on chlorophyll content in mung bean yellow mosaic India virus infected plants. As the leaves become yellow in this in yellow mosaic disease, there is an adverse effect on the chlorophyll content of the plantum. Plant extracts treated plants record a significantly higher content of total chlorophyll content than control. Treatment T3, that is Datura stramonium at 20% concentration, showed best in increasing chlorophyll content. Treatment T6 then content treatment recorded treatment T16, that is control, recorded significantly lower amount of chlorophyll compared to plants in the treated plots. Next. Impact of phytoextracts on photosynthesis and transpiration of infected plants. Increase in the chlorophyll content also increases the rate of photosynthesis and rate of transpiration also. So the rate of photosynthesis and rate of transpiration was also recorded higher in Datura stramonium at 20% concentration than control. Next. Impact of phytoextracts on yield in mung bean yellow mosaic India virus infected oat bean. Increase in chlorophyll content in treated plants and increase in the rate of photosynthesis and transpiration results in the increase in the yield. Maximum yield was recorded by treatment T3 Datura stramonium 20% concentration, followed by the treatment T12, that is Azadirachta indica at 20% concentration. Inference. Use of phytoextracts are influential in reducing virus incidence. Datura stramonium at 20% concentration was found to be highly effective in reducing disease incidence by 48.71% over control, followed by Azadirachta indica at 20% by 47%. Total chlorophyll content and photosynthetic rate and transpiration in the mung bean yellow mosaic India virus infected plants treated with Datura stramonium was significantly increased compared to control. Increase in chlorophyll content and photosynthetic rate due to phytoextract results in higher yield in the same treatment. Eco-friendly strategies are very fruitful in replacing hazardous chemicals for managing yellow mosaic disease. Thank you.
Thank you for the nice presentation, although it took some more time. But
sorry for that, sir.
No problem. Anybody else is there or not? If someone there, they should be prepared for presentation. Otherwise,
yeah, Mr. Vaibhav Pratap Singh is left. Is he there or not? I think he is not there.
Okay. So
we bind up it.
So
Dr. Mishra ji.
Only four presentations were there in this, and I think everybody has justified and within the time limit also, and there were presentations. I feel that this session is very important, and I will like to thank the IPS for the opportunity they have given me, and moreover to the President, Dr. Chakravarti, and Pratibha Sharma and Sri Mandal for nicely participating and conducting this session. I think it is very important, and young participants should be encouraged for this type of activities. I think the role of our Dr. Chakravarti and Dr. Pratibha Sharma will be more in coming years, and I am thankful to all of them. Thank you very much. Dr. Pandey, please.
Yeah, thank you, Dr. Mishra ji, Chair of the present session. Respected Dr. Chakravarti ji, Madam Pratibha ji. This is really this session is very important. Is APS travel grant is meaningful just for the shaping the career of the students. They will be going to present to IPS to the US and in American Pathopathological Society. So that is very good platform to shape up the career of the students, and also very good grant. And all the contestants, they were very good. They have done very good efforts to make the good slides and good work and present over there. And well in time, as told by Dr. Mishra ji. So this is very good neck to neck competition, and then the authorities will decide about the grant to the best candidate about it. And I'm grateful to Dr. Chakravarti ji for giving me this opportunity to share very important session, and also Professor Pratibha ji for and Madam Rashmi Agrawal, and I see here Dr. Pandey and everyone. So I'm thankful to IPS and Dr. Mandal for helping
Yeah,
us in this session.
I also see that and Dr. Rashmi is also there, and
Yes, I was listening partly in between. A lot of work was there.
Dr. Rakesh Pandey and Krishna Kumar are also there.
Achha, Rakesh Pandey is
He
there.
is also there. So thanks for all participation to by the all. We are thankful to all.
Yeah, thank
you. Thank you, sir. Thank you,
Dr.
Dr. Pandey, Dr.
Doctor.
From my side also, Dr. Kalyan, I will also thank profusely Dr. Pandey, Dr. Mishra, and Dr. Pratibha, madam, and Dr. Rashmi, Dr. Rakesh Pandey, and many, because this is one drawback of our virtual system, that we cannot see everybody at one. And
Yeah,
of course,
yes,
Dr.
certainly, sir. Yeah.
Secretary and vice. Dr. Sesha is also there. I'm seeing, and many of you are there. Who are very well conducting this seminar or this session. And Dr. Rashmi is always there. I know behind, even if he's not shown up, I know she has a double charge there and very multiple charges.
Yes, a very busy person.
Sir, this is really hard at this stage. It's a closing, financial closing, and a lot of issues.
Yeah, that is also coincidence that financial year is good.
Our staff, Dr. Mishra and all, they have initiated this kind of program. So
the
that
seniors support.
This opportunity, and all those who are involved with it in selecting and sending some of our very good students for getting their exposure. Nevertheless, it is not adequate because there will be so many other people. So with the help of subsequently, with our having more resources, so we will be able to have at least 5 students or 10 students subsequently in subsequent years to go there.
Yeah, sir. This is the beauty of this IPS family, that everybody is working in very coordinated manners and in the guidance of you all elderly people. So that is the beauty of this society.
Advancement, and subsequently try to manage some more resources. We are always there with her,
yeah,
so that
certainly.
we can facilitate more and more students, so that our country also progresses and advances.
Wholeheartedly, we are there for the society.
Thank you, sir. On behalf of
Thank you for your kind words, sir.
And on behalf of organizing committee, I extend my heartiest thanks to both the chairs and for nicely conducting these sessions. And this is a very important session that APS Travel Grant we instituted, and under the regime of one of the chairs, Dr. A.K. Mishra, he mentioned, I forgot to mention in this introduction. And I think we enjoyed the nice presentations by all the awardee, means all the contestants. And as per rule, you all know that it will be prioritized and all the name will be sent to the APS for APS grant. And finally, the selection will be visited on them by APS people. So I am also thankful to the Honorable P.K. Chakraborty sir, our Madam President-elect Pratibha Sharma ji, our Madam Head Madam Dr. Rashmi Agrawal Madam, and all the dignitaries. I am again limited to the only screen. So we are really thankful, sir, to both the chairs and all the speakers and all the dignitaries. Thank you very much.
Yeah,
But
thank you.
before concluding, I have one important announcement that we are rescheduling the next annual meeting, general body meeting, that will be instead of 2 p.m. We are going to start by 2:30.
Okay.
And 2:30 to 4, it will continue 1.5 an hour. And then 4 to 5, we are having valedictory session. So all are requested to join in the valedictory session. Thank you very much.
Thank you.
Thank you. Thank you all.
Thank you all.